Interactions mutualism competition session. And as we all know, species interactions underlie much of the living world as we know it. And today we have a great ledger of a variety of talks going from classic pollination, mutualistic interactions, all the way to coevolutionary arms race and competition. So without much ado, I would like to invite our first presenter, Lucas Camacho, who's going to talk to us about coevolutionary exploitative interactions and how they can increase trait disparity and modularity in mutualistic networks. I'm going to mute myself now, and you're on, Lucas.
Okay, and thank you for the introduction. I will share my screen now. Okay, as everybody see my presentation? Nice. I will just start here a second. Okay. So thank you everyone for having me here today. It's a great pleasure to talk to you all. And I'm here to talk a little about my master's project, which I did in collaboration with several other researchers who helped me a lot in my first academic experience. So I will today talk about a brief introduction about what was my master's project. Then I will say about the coevolution model that I used to answer my questions. Questions about the frequency of exploitation and coevolution, and the structure of networks and exploitation. And finally, I will give some final and general conclusions. And I would like to start this talk talking about this incredible and diverse interactions that is the mutualisms that you are seeing in the slide, the pollination, the seed dispersal, and the anti-Myrmecoff interactions. Of course, there are other several types of mutualisms, but here in this talk, I will focus on these three types. And we can, mutualisms is all about the benefits. So we can describe it as interactions which both of interacting species are benefited. But of course, mutualisms is not only about benefits, it's about cost as well. For example, we have the cost of seed and fruit production for attract possible seed dispersers. We have the cost of product nectar and pollen for attract possible pollinators. And we can think about in another cost related to the trait matching, which is the similarity between the traits mediating interactions, for example, between these two species here, the bill of the hummingbird and the tube of the flower. And maybe this trait matcher could lower the potential of other interactions that these species could have in the community. So we can think this as a trait matching as a cost as well. And thinking about these costs, maybe mutualisms are prone to be explored by other species who doesn't contribute to the balance of the costs and benefits. We have some examples in here. For example, this bee who would not touch the reproductive structures of the flower. She makes a hole in the base of the tube, gets the nectar, and doesn't pollinate the flower. Or for example, some mammals who does not only eat the fruits, but also eats the seeds of the plants. Or some examples of ants in Cecropia, which do not repel some herbivores efficiently. So we can take these interactions as interaction which one species obtain the benefits of mutualisms without providing benefits in exchange. We can take these interactions, mutualisms and exploitative interactions as a way of negative and positive effects. For example, that first example of the bee and the flower, we can describe it like this, a positive effect from the plant in the bee and a negative effect from the bee in the plant. And we also know that there are not only pairwise interaction in the communities. Actually, there's a lot of interaction going on at the same time, and so in different interactions. So put it in a network context, we have something like this, a bipartite network with mutualism and exploitative interactions happening at the same time. So thinking about that, we can describe positive and negative effects in interactions. And you know, there's a lot of interactions going on, as we can describe in networks. And also that these interactions will change how species are reproducing and surviving. So we can say that these interactions could influence coevolution, the reciprocal evolutionary change between interacting species, and coevolution will change traits and how species are interacting in the community. So we have a feedback between ecology and evolution here, networks changing evolution, coevolution changing the interactions. And thinking about this, my main question is how these exploitative interactions that I showed to you will drive the coevolutionary process in mutualistic networks. For that, I will explain a little about the coevolution model that I use to answer some of these questions. This model, I try to describe a single trait from a species, a population of the species I. For example, here we have several individuals with different traits of species I, and I can get the average trait, which I call Z. And this is the trait that I will describe in my model. And how will I describe this trait in my coevolution model? Well, this trait depends on the interactions, the mutualistic and the exploitative interactions, mutualisms in blue and exploitative in red in here. We also have all other selective pressures not directly related to these interactions that I call here the environmental selection here in black. And I also need, is the coevolution model, so I also need take into account the selective gradient of selection, which is the minor change in traits changing the fitness, the average fitness of this species I, and the additional additive genetic variance of the species. So we have a model like this. We have our components of interactions in here, and this parameter phi, which takes into account this additive genetic variance and the selection gradient. And I also have some assumptions related to this model, which how the dynamics of mutualism and exploitative interactions happens. Oh, I will show you some of this right now. For example, in mutualisms, we have these traits, the bill of the hummingbird and the tube of the flower. And here I have the trait distribution of the community. And I expect that selection will favor trait matching for the coevolution of mutualism. So in time, I expect that these traits will become very similar. For exploitative interactions, it's not, it's kind of it's a different. We can consider the same traits in here, and now they are interaction in an exploitative way. And I expect that selection favors for the exploiter species trait matching. So individuals who are who resembles the victim will reproduce and survive more. And but for the victim, we expect trait mismatch. So individuals who are very different to the exploiter will survive and reproduce more. So selection will favor this type of dynamics, like an arms race dynamics. So we have these two dynamics depending on the interaction. Okay, I have this coevolution model here, but I also need to know which species are interacting in the community. For that, I downloaded some empirical networks from the internet. I downloaded 8 networks of anti-myrmecotic interactions, 8 of seed dispersal, and 8 of pollination from these 2 databases, Web of Life and IWDB. I'm very lucky to have this data available and online for free for download. So it's very nice. So now using my coevolution model and using these empirical networks of mutualisms, I write a code, a computer program. And this code is available on GitHub. If everyone would like to see and criticize me, it will help me a lot. And with this code, I, like magic, will have simulations of how this mean species traits change in time. This was supposed to be a GIF, but I think it's not working. How this mean species traits of each species in the community will change due to the coevolutionary process of mutualism and exploitative interactions. So now I have my framework, and let's start to answering my questions. So just a second to water. All right. So my first question is related to the frequency of exploitation and coevolution. So we can think in a network, we can have several frequencies of exploitative interactions. For example, here we have an axis, and in the left side, we can see there's just a few, actually just one exploitative interaction, and all the other ones are mutualisms. And as we move to this axis, we can see that the frequency are increasing. There's a lot, I believe that every interaction in this right network is exploitative interaction. So how these different frequencies of exploitative interaction will affect the coevolutionary dynamics? For that, I will use numerical simulations. I will use my 24 networks that downloaded on the internet, and I'll explore frequencies of 0.01 to 1 of exploitative interactions. And I will do 3,000 simulations per network in a total of 72,000 total simulations. And what I expect that due to that arms race dynamics that I showed to you, I expect that in higher frequencies of exploitative interactions, there will be a higher trait disparity. So the mean distance of traits of species in that community will be higher, species will be more different due to that arms race dynamics. But also I have a component of trait matching happens in here, because some other mutualism may be happen in the community, or there is that component of the exploiter who still selection favors trait matching for him. So I do not expect only trait disparity, but I also expect higher trait clusters formation, which are species traits that are very similar to each other, like a group formation of traits. And after I did my simulations, I saw that in networks with higher frequency of exploitative interactions, there is a higher trait disparity for all the three types of mutualisms tested. But when we saw the group formation, the number of trait clusters, there is an initial increase of these trait clusters due to the frequency of exploitative interactions, but there's also a decrease. So we're not exactly expecting that. And what I see in my results, and I think that's the main contribution of this first question, and this maybe could be a possible explanation for low trait matching empirical systems. Pedro Giordano shows in his amazing work that these different seed dispersal syndromes in here. So maybe what's happening is not only trait matching due to mutualistic interactions are molding species trait, but also other types of interactions, for example, this negative effect from exploitative interactions. Also, it's very, very curious that the same pattern could emerge due to different processes. This incredible work by Lucas Fernandes and his collaborators shows that when you link mutualisms and antagonisms in space with gene flow, we have the same pattern of trait clusters formation. So maybe what's happened there, there's more than one process, not only gene flow, connect interactions, but also different interactions in the community are maintaining this kind of process. And finally, I'm very curious about this linear of exploitative interactions and how we can test this in the wild. Now that we made some numerical simulations, maybe it's time that we can go to the natural communities and try to some way see how much of this, how much of frequency of this exploitative interactions happens in natural communities, and how is this translated in the trait disparity of natural communities. Okay, I will move to my second question now. It's about the structure of networks and exploitation. So we know that there's two very known basic structures in mutualistic networks. Modular networks, which are more compartmentalized and generally are less connected, is very common in ants, plants, and networks. And we also have these nested networks, which are highly connected generalist species with specialist species interaction with those generalists. So it's common in pollination, in seed dispersal. And we can think now that there is a component of trait disparity who is increasing trait disparity in the simulations. So what happens if maybe these species are becoming so different, but so different that they cannot interact anymore, like a trait barrier? How the coevolution process of these different types of interaction will change the structure of the network. So for that, I will do numerical simulations again with my 24 networks. I will explore the frequencies of 0.01 to 1 of exploitative interactions again. And I will do my 3,000 simulations per network in a total of 72,000 simulations. And what I expect is that Due to this higher trait disparity from the exploitative interactions co-evolving mutualisms, I expect that the networks will become more modular and less nested in higher frequency of exploitative interactions. And after that I did my numerical simulations, I see that this pattern of increasing modularity in decreasing nested is seen in all three types of mutualisms. In the ant-plant networks, this pattern was not so strong, maybe because this networks is already modular. But we can see here that this increasing modularity and decreasing nestes, these black points are parallel simulations, which I remove interactions randomly just to compare how is the loss of interactions due to coevolutionary process and the random loss of interactions. We see that there is a changing structure for both the black points and the color red dots. But maybe there is a strong, how can I say, changing structure when we consider not only loss of interactions randomly, but loss of interactions due to trait disparity.
Lucas, you have 4 minutes left.
Thank you. Now, maybe this is loss of interaction and increasing modularity was already seen by Cecilia Andreazzi and his collaborators, which show that in antagonistic networks, where the loss of interactions will also increase the modularity and decrease nested of antagonistic interactions. So maybe what we see here is a path of mutualistic interactions networks becoming more and more like the antagonistic networks when we increase the exploitative interactions and the coevolution process happening, changing traits and species lost in their interactions. And finally, I'm very curious about what will happen if we do not lose only interactions, but also we lose species. How the coextinction simulations will, how the different coextinction simulations will disrupt the network, and how much of the exploitative interactions we will need to completely disrupt a network. So I'm very curious about that. So I will make some general conclusions about my work. We see that exploitative interactions generate trait disparity and increase with the decrease of number of species trait clusters in mutualistic networks. Also, we see that exploitative interactions generates more modular networks than random removal of interactions. And finally, I'd like to thank all of my collaborators who helped me a lot in this process, all the past and present members of the Miudo Lab, CNPq for funding my master's degree, and the Department of Ecology at USP, all the staff of the virtual Iamara, and thank you for listening to me talking for more than 5 minutes. Any doubts, you can talk to me now or just send me an email. Thank you a lot.
Thanks, Lucas, for the fantastic talk. And we definitely have a couple of quick minutes for some questions, and I'll let Peede take that over.
Just give people a minute if they're typing questions, either in Slack or chat. I'll just relay Judy's question. I assume your model used very costly exploiters, but a lot of exploiters have fairly weak effects. Would it be possible to use your model to see if the effect of exploiters on coevolution increases linearly with severity of exploiter effects? I could imagine alternatively that there's a threshold below it would be there would be no effect, and above it there will be a high effect.
Actually, my model considers the effect of mutualism and exploitative interactions very similar. So what could be done is that changing how the dynamics of the model works. So in the part of the dynamics in which there are exploitative interactions and the trait matching mismatch, this I could change how this dynamics works and put some asymmetries maybe in how this interaction are strength, right? So maybe we can lower the effect of exploitative interaction, see what happened, of course. Yeah, it is a assumption of my model. The effects are very similar. Yeah.
That's cool. Maybe we have time for one more question. John's asking, can you partition the effects of direct and indirect effects in your networks?
Well, I think yes, but I didn't do it. I am very curious about that too, because I think that uh, if mutualistic interactions are so important for the indirect effects, maybe we have a other force for the other side. Maybe what holds these indirect effects in mutualist networks, if we're considering exploitative interaction as well, is the effect of exploiters. But I'm very curious about that, but I didn't do it yet.
Okay, thanks for that, Lucas. Thanks,
Yeah,
Casey.
thanks.
There's more questions that I'm sure you'll be able to answer later on in the chat or directly to the people.
Thank you.
We'll move on to our next speaker, just keeping an eye on the clock. And we've got Joshua Mesa, who's going to tell us about the evolution of increased tolerance in the invaded range of the yellow star thistle. Joshua, if you can share your screen. Yeah, it's good. It's over to you now.
All right, thank you very much. Okay, so yeah, today I'm talking about the evolution of increased tolerance to herbivory in invaded range yellow star thistle. And again, I'm Miles, and this is work that was conducted in the lab of Karina Dlugosh, who was my postdoctoral advisor. So starting out, this is with invasive species. And a couple of things that we know about invasive species are that they are a major component of global change, and they have a wide range of economic and societal effects and impacts. And they cost more than $120 billion in damages and in control efforts. And they threaten all kinds of different industries, whether that's agricultural, forestry, and fishing. And they're really a major problem and threat to people's livelihoods, especially in developing countries. But we also know that they have a large ecological impact as well, as they can outcompete native species and displace them, which can lead to species extinctions and a loss of biodiversity. And they can completely alter the community structures in which they invade, which can degrade ecosystems and impact all kinds of natural cycles, such as the nutrient, energy, or water cycles in that area. So one of the major questions, though, is what leads an introduced species or an exotic species to become invasive? And there's a lot of hypotheses that have been explored to account for invader success or invasiveness. And the rapid evolution of an invasive species and its traits is often invoked as a key to a successful invasion. And there's abundant evidence that novel species interactions play a role, whether that's from the loss of competitors or mutualists and changes in plant enemies and the herbivores. And it's really essential to kind of understand these different factors so we can help facilitate a successful control of these different kinds of species, understanding why they're becoming invasive in these areas. So native and invasive plants interact, of course, with a wider range of different kinds of herbivores, and those herbivores within the community can vary greatly. And it's really often thought that invasive species are able to experience a relaxation of enemy pressure in their environment or in their new novel environment, such as like enemy escape. So that way they can focus less on plant defenses and more on increasing growth and reproductive potential. However, there's some studies that suggest that they are still actually susceptible to their novel generalist herbivores in their new environment. So if we look over here from this one meta-analysis, we have, can you see the cursor? I'm going to assume that you can. We have plant origin, which is we have native plants and exotic species coming into this native area. And we have herbivore effect on the y-axis. And a negative number here denotes that it's a negative effect on the plant from an herbivore, from these native generalist species that are chomping on them. So what we see here is that actually exotic plants have a more of a detrimental effect from these native generalist herbivores, which they're just now encountering, than do the plants that are already residing there. which is kind of interesting because, you know, one would expect that relaxation of enemy pressure, but in some instances, it looks like there's actually an increase perhaps, or at least, you know, that they're getting hammered more than native plants. So that kind of begs the question, you know, what's going on there, and what is exactly evolving in these areas? And how can they interact with these herbivores that they're encountering? One of the major defense traits for plants is, of course, plant resistance. These are pretty, you know, typical what people know about. There are traits that prevent or reduce tissue damage, and they can be structural traits such as spines and thorns, or trichomes like over here. But then they can also be chemical defense traits such as secondary metabolites, like, you know, glucosinolates or tannins, something that reduces metabolism or something like that, or is toxic to an insect or a mammal. And most studies of invasive species and their natural enemies have focused on plant resistance. However, there's also other types of interactions that a plant herbivore can have, and this is, or that a plant can have to herbivores. And that's namely plant tolerance, which is a regrowth strategy. And this is basically a compensation for any tissue that is lost through damage through herbivory. And there's been recent recognition that plant tolerance may actually play a role in facilitating invasions. But there's still comparatively few studies when you look at, you know, compare tolerance to resistance. And we think it could be an important, given there's so many novel journalist herbivores that are still damaging these plants. So maybe that there's been an increase in plant tolerance in some of these invasive species, allowing them to cope with that increase in damage. And this is just an example of you know, what plant tolerance or compensation can look like. On the left part of this photo, we have a just typical plant here. Something comes by, like a deer or something, eats it, chomps it all the way down to the base rosette, and then it can regrow. And in this case over here, it has more branching number and kind of has more real estate allowed for flowers and seed production. So in this particular case, this plant actually benefited from being eaten. So that kind of alludes to this, that tolerance can kind of exist on a continuum. And this is some data from Arabidopsis, some different ecotypes, showing that over here on the left-hand side, on the x-axis, we have, you know, 3 ecotypes here in these gray bars. And we have percent change in seed production when you compare damaged versus undamaged plants. And these 3 on the left actually do worse after being eaten. They are able to regrow, but they still are not able to fully compensate you know, for the damage that they incurred. Whereas in the middle, these 4 in the white bars, they're able to fully compensate after damage, and they're able to produce just as much flower and seeds as their undamaged counterparts. And then over in the black bars here, we have a few ecotypes that are able to increase their seed and fruit production after being damaged. So they overcompensate. So tolerance kind of exists in an entire spectrum that we can look at. And our question really is, how has plant tolerance evolved in invasive rangeland pest species? So for our study, we used yellow star thistle. It's a very highly successful invader, especially in Western California and the Western United States. And it's native to Eurasia, but again, it's been introduced to both South and North America. And the invaders show increases in size. So if you look over here at this picture, we have a native ecotype over here, and then from an ecotype from the California invasion. And the plants are not only show an increase in biomass, you can tell it's much larger, but they also have an increase in reproductive potential as well. And there's some evidence of changes in how the immune response is working with, you know, some types of pathogens. But one thing that we do know is that yellow star thistle interacts with hundreds of species across its invaded range. However, there's a lack of information on how plant tolerance may be changing. So that's what we really wanted to look at. Yeah, so the question is, how do native and invaded populations of yellow starthistle respond to damage? And for that, we took 4 invaded range populations from California, and we also had 4 populations from Western Europe as well. So these are native ones, of course. And then for our design, we used a randomized blot design in the greenhouse. And once these plants, really, they set the rosettes, and then they started to elongate their inflorescence. Once they started bolting, we went in there, damaged them, removed the inflorescence once they reached about 10 centimeters of height, and cropped it down to where only the rosette is left. And we did this because in this area, they're often mowed or grazed as a form of management and control in California. So that's why we did this particular one instead of, insect damage and like rosette damage. So then we took both our control undamaged plants and damaged plants and allowed them to go and set flower. And then we went in to take our biomass and reproductive fitness as well measurements. And this was to assess both the vegetative and reproductive tolerance of these plants. So what we have here is what we see is that if you look over here at our native populations on the left, we have capitula number or flower number on the y-axis. We have our control plants that were undamaged from the native range, and they significantly do worse after being damaged. They undercompensate. So they're not able to fully compensate and recover after being damaged. However, plants from the invasive range are able to equally compensate. They're able to just, do just as well as their undamaged counterparts after being damaged. And we see similar things with vegetative compensation. And this is from biomass measurements. So basically went in, threw them in the oven, take their biomass. And we see similar things. Native populations are smaller than their, after being damaged, whereas the invaded range are able to fully compensate. So it does look like that the invaded range populations have evolved an increase in its tolerance or compensatory ability during this invasion. And one of the other things that we wanted to do, and I was interested in, was kind of exploring any type of mechanism for this increase in tolerance. And one of the reasons I was really interested in this is because the lab that I was previously in, we did a lot of work with plant compensation and understanding what kind of mechanisms leads to increased compensatory ability. And we did this in Arabidopsis. And what we found was that this process of endoreduplication was the causal mechanism for plant compensation. So in case you don't know what endoreduplication is, it is successive rounds of mitosis without subsequent cell division. So after a couple of rounds, you go from like a 2N ploidy, and after a couple of rounds, you just keep going up until like 16N. You never divide afterwards. So you can very quickly go from like 10 to 80 chromosomes. And this leads to different kinds of nucleotypic effects, larger cells, and you can also have increases in gene expression and things like that as well, and increases in water transportation between cells. So just a little bit of background on it. This is from Arabidopsis. We have this black circles here is an ecotype Columbia, and this is able to increase or overcompensate after being damaged. And if you look over here at nuclear DNA content after being damaged, it has a significant increase. Whereas this Landsberg ecotype is an undercompensator. It doesn't change, has no ability to undergo endoreduplication. And then if you expand that out with a bunch of different ecotypes, we still see a general relationship between the two. So we have change in cell cycle value, which is our measure for endoreduplication. So over here is very high, and up here as well, we have on the right-hand side, increases in seed production after being damaged. So there's a generalized relationship, and this was followed up with genetic manipulations actually showing that it was a cause and effect between the two. So this is an undercompensating ecotype. It is a different Columbia than the one I showed earlier, but this one, after being damaged, has a reduction in seed production, as you can see in the white bar over here on the left, as well as biomass. And it has a negative cell cycle value after damage, so it does not endoreduplicate. And whereas if you increase endoreduplication experimentally via this overexpression of increased level of ploidy-1, you not only increase endoreduplication levels, but you also change this plant from an undercompensator to an equal compensator. So yeah, that was kind of some background on why I was interested in this and some of the work that was done previously. So we wanted to do the same thing. And to do this, we use flow cytometry. And one of my undergrads helping me with this that really focused on this was Jesse. I unfortunately don't have a picture of him on my computer, so we'll just say this is Jesse. He's a very happy guy. And very helpful for this whole thing. So anyways, you just put in your cells more or less after you chop them up, release the nucleus, you hit it with a light source and it measures fluorescence, so you can get like your different ploidy levels. So we used Arabidopsis for reference, just kind of showing you this is the kind of graph. So this goes all the way up to 32N. And then taking, now looking at yellow Starthistle, we just didn't find anything. It just is 2N. And, we put a lot of work in, and it just turns out that yellow Starthistle just doesn't endoreduplicate. So that pretty much shows that endoreduplication is not, the only thing that can lead to increases in compensation. One of the things that we really want to look at is reallocation of resources from the roots and different things like that. Some of the work that's been done in the past as well in other species. But yeah, anyways, so we found, again, just as a conclusion going over things, rapid evolution of invasive species is often invoked as a key to a successful invasion. And plant tolerance may play an understated but important role in invasions. And it could be really important for the types of management we use for these plants that are able to compensate. Because as I said earlier, yellow Starthistle is often mowed or grazed for management. So that could really pose a problem if you go through, mow it, and it just pops up and it just has no fitness effects. It's able to produce the same amount of flowers and seeds. And it can also have possible implications for long-term biocontrols if it increases in the tolerance as we have shown. And of course, endoreduplication was not the mechanism for this increase in tolerance, but there's some, I found this paper that I thought was interesting, looking at the yellow Starthistle's deep root system, and they were seeing that reallocation of resources, or what appeared to be reallocation from that root system, makes them very mow regimes, which, so again, they were experiencing these difficulties with mowing to control it. So, yep, that's all I got. Some acknowledgements from everyone in the lab, and of course, my excellent undergraduate research assistants, Jesse and Michaela. Michaela's work is different, so it wasn't here, but yeah, that's all I got. So any questions, and thank you.
Thanks, Joshua, for that excellent talk. We have time for maybe one very quick question.
Oh, sorry, didn't know how long it was going.
Hey, Miles, I'll pass on a question from Slack from Ellen. Ellen Sims. She says, cool study. Flowering time could be a tolerance mechanism, especially if mowing or herbivory is uniform across individuals in time. So why did you choose to clip at 10 centimeters rather than at a single time? And how might the results be different if you'd done it the other way?
If we, oh, okay, so okay, I see what you're saying. Yeah, you can definitely, I think, we just wanted a uniform type of, more or less, because that is true. Some of them flowered earlier than others. And if we clipped everything at the same time, sometimes we weren't going to even have anything but a rosette at that point. Some of them were definitely taking longer to actually, mature. But that's a good point. And, changes in phenology can definitely be a way to avoid any types of damage as well. So that's a whole other, thing that you can look into for avoidance of herbivory completely instead of like tolerance or compensation instead. This is what we were interested in, or what I was. So, yeah. But that's a very good point.
Yeah, I don't think there's a right or wrong way to do it. You're missing something whichever way you do it. But
yeah, definitely.
Yeah.
All right. Thanks,
Cool.
Josh, for that. Thanks, Casey. Just keeping the time.
Sure.
We're going to move on to our next speaker, Joice, and she will tell us about the evolutionary inferences on the community organization of flower-hummingbird interactions. Joice, we can see your screen now, so it's over to you. Can you please unmute yourself, Joice? Joice, we're not able to hear you. Looks like there might be an issue with your audio settings.
Do you hear me now? Yeah, okay. Sorry. Okay. So I'm going to present this work that I developed together with Gabriela Glazer, Gustavo Burin, Jimmy Maguire, Marcelo Eisen, and Silvana Buzak.
Sorry, Joice, can you please share your screen again?
Okay. Okay, sorry. Well, so there are a lot of factors that may influence the structure of communities, and one of those factors is pollination. So pollination might influence the community structure by several ways, for instance, promoting competition or facilitation, and influencing the species distribution or species coexistence. And the way in which pollination will interfere on the structure of communities will depend on the degree of dependence of interactions. In hummingbird pollinated systems, the interactions tend to be very specialized, especially for some plant groups like Heliconia and Bromeliaceae. But when we look at the hummingbirds with more attention, we see that there are a lot of differences between the species that allow us to classify them into two ecological groups, the hermits and the no-hermits. These differences are related especially with the morphology of the bills, with the feeding behavior, the degree of diversification, and the geographic distribution. And in general, the hermit hummingbirds tend to be more specialized than the non-hermits. And the morphology of the hummingbird and all the other species are associated with the morphology of the flowers. And when we have the straight matches, these interactions appears as a modules in plant pollination networks. A module is when an interaction tends to occur more often than others. And these interactions are probably a result of a joint evolutionary history. And when we have a joint evolutionary history, like coevolution, the phylogenetic tree of the groups tends to mirror each other, like what happens in gophers and lices. Obviously, for pollination, we cannot expect a perfect congruence, but we still can search for phylogenetic signal between the phylogenetic trees. This signal suggests that the current association are a product of a joint evolutionary history in which ancestors share similar evolutive pressures. And so in this study, we intend to answer two questions. The first one is if the cophylogenetic signal changes between communities pollinated mainly by hermits and non-hermit hummingbirds. And what we expect is to find that a higher cophylogenetic signal for communities pollinated by hermits rather than non-hermits, because this group is more specialized. And the second question is if the cophylogenetic signal in communities visited by these two groups of hummingbirds changes with latitude and altitude. And we expect to find a higher cophylogenetic signal in lowlands and areas close to the tropics than for highlands and areas far from the tropics, because hermit hummingbirds, the more specialized hummingbirds, are restricted to lowlands and areas close to the tropics. So to answer these questions, we use two phylogenetic trees, one for plants and one for hummingbirds. So in this phylogeny, we can see the hermits here. They are represented by the hermit clade, and they are older and less diversified than the others. We also used two analyses. The first one is Procrucis' approach to cophylogenies, also known as PACO. And this analysis yields a global goodness of fit statistic and a p-value, and it yields the most important associations, meaning that they show us the associations with higher contribution to the signal. The second analysis is complemental. It's called Handel dentagram partitions, and yields a normalized Gini coefficient. These coefficients ranges from 0 to 1, and is inversely proportional to the cophylogenetic signal. So when we have a value of Gini coefficient closer to 0, we have a strong signal, which means to say that every interaction contributes in a unique way to the congruence. So every interaction is unique. And when we have a value close to 1, we have a weak signal. That means to say that every interaction contributes in a similar way to the signal. We do have a threshold. This value is 0.72. For more detail, please see the original paper of the analysis. This threshold is 0.72, and below this value, we have a strong signal, and above a weak signal. The interaction data, we use it, what we organized from a data paper on plant vertebrate pollination in Atlantic forest that will be submitted very soon. And we divided this data into 8 latitudinal ranges and 5 altitudinal ranges. Each one of these ranges is considered as a community. So we have 8 latitudinal communities and 5 altitudinal communities. And we calculated the beta diversity for among these communities, and also the cophylogenetic signal. So we ran PACO and random tapas. First, consider the hummingbird group. So we ran this analysis for the whole data that we called MetaWeb, and then we divided into 2 groups, the hermits and the non-hermits. And then we did the same for the 5 altitudinal communities and for the 8 longitudinal ranges. And we ran this analysis in a total of 33 times. And our results is this. Well, here we have the phylogenies of plants on the left and for hummingbirds on the right. The lines represent the associations. Blue lines represent associations with higher contribution to the signal, and the gray lines represent the associations with low or non-contribution to the signal. And what we can see is that we did find a congruence, a cophylogenetic signal for all cases. You can see here the p-value was equal to 0. Which is expected since these interactions are very specialized. But when we look at the Gini signal, Gini value, we see that we find strong congruence only for the MetaWeb and especially for non-hermit webs. And we didn't find a strong signal for the non-hermit web. And this is also expected since the hermits are more specialized and probably had a joint evolutionary history. We can see here that is a group of plants with higher contribution to the signal. These are the bromeliads. And we also have a group of hummingbirds with high contribution to the signal. And these are the hermits, and especially one species named Nymphodon. Regarding latitude, here we have the results of the diversity. Here we have the beta diversity. Then the turnover rates, which represents the change in species composition among communities, and the nestedness that indicates the loss of species among communities. And what we had is a higher value of biodiversity and turnover, and a low value of nested for both plants and birds, which means to say that the communities in Atlantic forest are highly diverse and rich. Regards to the cophylogenetic signal, we remember that we hope to find a higher cophylogenetic signal for areas closer to the tropics. In this plot, we have the results, the Gini value, for each one of the latitudinal range, and the dots represents the communities. So here we have the meta web, the hermit web, and the non-hermit web. And the fillet symbols represents the communities in which we found a congruence, a significant congruence between plants and hummingbirds. So we have to look for these points. And we did find a strong cophylogenetic signal for some communities, so the Gini values below 0.72. But in general, when we did find congruence, was in the areas, in the tropical areas and not in the subtropical areas. For the altitude, we found similar results for plants, but for birds, we found low turnover rates and high nestedness rates, which is also expected since we have some loss of species with elevation. The hermits are restricted to lowlands. And we hope to find that Gini coefficient, meaning that we hope to find a higher cophylogenetic signal in lowlands. And here we have a similar plot with elevation. And we didn't find a strong cophylogenetic signal for any case, but when we did find a congruence was in the lowlands. Well, comparing these two groups of hummingbirds, so we found evidences for a joint evolutionary history for hermit hummingbirds. These hummingbirds are more specialized in morphology and trapline behavior, and especially with bromeliads. But when we look, we cannot use the plant phylogenetic tree that we have because it's not dated, it's not calibrated. But here we have a phylogeny that's calibrated and with the reconstruction of the pollination evolution for the bromeliaceae. And what we can see is that the orange of this group was from 20 million years ago. And it coincides with the orange of hummingbirds. More especially, the orange of that hummingbird with higher contribution to the signal, the Humphodon nevius, was 15 million years ago, and it coincides with the orange of bird pollination in the bromeliads. This is another evidence that maybe this group of hummingbirds had a joint evolutionary history. They shared some similar pressure with these plants. We didn't find the same results for the non-hermits, and they are very generalist hummingbirds in territory, in the distribution, in the foraging behavior, and also in biomorphology. So here we have some examples of hummingbirds with a great variation on biomorphology. And we also saw that a high diversity and a high species turnover in the communities in Atlantic forest. This might influence the spatial distribution and the phenological overlap and the trait matching on these communities, which, depending on the ecological context, might force the non-hermits to behave in a more specialized way, and then organizing these communities in some places. The evolutionary context of the communities also may force these hummingbirds to behaving in a more specialized way. Here we have an example of this species, Tallurania watertoni. It's a non-hermit hummingbird, and it has a trapline and a territorialist behavior, and it's restricted to the northeast portion of the Atlantic forest. And when we look at the results that we found in this area, we did find a strong phylogenetic signal. And this hummingbird, together with another hermit hummingbird, contributed more significantly to the signal. So in this case, we might also have evidences for joint evolutionary history of these hummingbirds with the plants, especially the bromeliads, in this area in some communities.
You have 5 minutes.
Okay, thank you. In conclusion, we found evidences for a couple of evolutionary history for hermits and plants. We didn't find the same for the non-hermit hummingbirds, but depending on the context, the evolutionary and ecological context of communities, these very generalized hummingbirds might behave in a more specialized way and then organize communities. And we also found evidences for co-phylogenetic signal in tropical regions and in lowlands. These works have some caveats, and one that I would like to point out is that we used an uncalibrated phylogenetic tree for plants, which makes comparisons very difficult to make, and also it makes difficult to draw some conclusions. I would like to thank the University of São Paulo, Department of Ecology and CNPq for allowing this work to happen, the American Society of Naturalists and Iamara for the opportunity to show my work, my co-authors, and some people that help with comments and data organization. Thank you very much.
Thanks, Joice. That was fantastic talk and great timing. So we have plenty of time for questions.
Giving people a few seconds to type out their questions. Joyce, John Thompson asks, are there plant taxa that show a joint history with hermits and non-hermits?
I'm sorry, I didn't quite understood.
Are there any plants that have a history with both hermits and non-hermits?
No, only bromeliaceae. Bromeliaceae are a very important group in Atlantic forest, and I think that they might avoid to find signal with other plants. I'll have to run this analysis without this group because I think that it might show something, but with bromeliads, we didn't find for another groups.
And then there's another question from Lucas Camacho on Slack. He says, nice talk, Joyce. Do you know how using a calibrated tree will change your results? So thinking that maybe the evolutionary history of this species are very important, maybe using a calibrated tree will reinforce your main results about plant hummingbird coevolution.
Yeah, it would also change a little bit of the analysis. So this analysis, the second one, random tapas, is a very recent one. And it changes the threshold, it changes the conclusions you can make. So it would be very, very different results if you had a calibrated tree, but I believe that it's a very hard task. I don't think that we have so soon because especially for Atlantic forest, it's very diverse place. But yes, it will change the results probably. Yeah.
Cool.
I expect that we would find stronger co-phylogenetic signal.
Okay. Any other questions?
Right, if there are no further questions, thanks again, Joyce, for the talk. And I will maybe invite our next speaker, who is Natasha de Manincor. And she's going to discuss the biases in our perception of plant-pollinator networks, which is a very intriguing title. Natasha, if you could share your screen. Yep. It's up now. And over to you.
Okay, thanks for the introduction. So, hello everybody, my name is Natasha, and today I will present some results of my PhD thesis that I did in France at the University of Lille about our perception of plant-pollinator networks with a comparison of visit and pollen-based representations of the same network. So, networks can be used to describe ecological communities and interactions among species, but in a context of climate change and species loss, it is critical to understand the mechanisms that drive network stability in order to preserve these networks. So it is important to have an exhaustive representation of ecological networks. In this talk, I will focus on plant pollinator networks with a major focus on wild bees. So most part of plant pollinator networks are based and construct from the observation in the field of the interaction between a flying insect and a flowering plant. But some of these interactions remain unobserved and probably giving a biased picture of the plant pollinator network. So an alternative method to study plant-pollinator interaction not observed in the field is the use of pollen, since it's a natural marker of the pollinator activity because you remain attached to the pollinator bodies after their visitation. So in these studies, I focus on 3 calcareous grasslands in 3 different regions in France, distributed along an environmental, but also a diversity gradient, because the southern sides display highest diversity. And in this size, I sampled recorded plant pollination interactions in the month of July 2016, which was one of the richest months of this year. So among all the sample insects, I focus on the female bees, and I collect the pollen from the insect bodies to prepare 2 pollen slides, one with the pollen collected on the specialized structure and one with the pollen scattered on their bodies. And I decide to separate the 2 pollen types because they bring 2 different information. The pollen scattered is the pollen passively transported. It gives a spectrum of all or potential old visited flowers, which are originating from visit for other type of resources, such as nectar. And this is the pollen also available for the pollination event. While the pollen collected is the pollen actively collected by the pollinators on a selection of plant species, is stored in specialized structures, and is the pollen that they bring to the nest to feed in the larvae. This pollen is often packed and mixed with saliva enzymes, so this partially reduces the pollen viability. To help us in identification of the pollen grains collected from the pollinators' bodies, I collect the anthers of all the flowering species in our 3 sites, which has an area of 1 hectare each, and we prepare 3 local pollen atlases. So in this study, we have 3 hypotheses. The first one is that the scattered pollen provides more information than the actively collected pollen. The second hypothesis is that richer communities are more likely to be undersampled than poorer communities. And the third hypothesis is that the addition of the pollen information can lead to changes both in the nectar structures and the species role, especially for singleton species. So to try to respond to our first hypothesis, we compared the rarefaction curves from the 2 pollen types, and we didn't find any significant difference in the number of realized links in any of our sites. So even if we find a percentage of unique links, which was higher for the pollen scattered as we expected. So since we didn't find any difference between the pollen type, we merge information in a 1 pollen-based network, and we prepare 1 pollen-based network for each site. As other studies point out, when we add the pollen information in a pollen-based network, we have a higher number of species and a higher number of links compared to the visit-based network. But we cannot compare these networks in classic network metrics because they are strongly affected by the nectar dimensions. So to avoid this problem, I create a simulated network where we fix the number of interactions per pollinator species, as in this example here, but we randomize the interaction pairs recording using the information in the pollen-based network. So now that we have two networks with comparable dimension, we can investigate where the addition of the pollen information can lead to changes in the network structure and species role. And to do so, I use different indices or methodology. For the nectar structure, I use the connectance, which is the proportion realized links over all possible ones, the H2 index, the index of network specialization, and a clustering methodology, which is the block model method. And to investigate changes in species roles, I use the species positions. In gray, there are other methods that I use, but I will not present today. So despite the fact we found marked differences in both species richness and also in the interaction both for the visit-based network and the pollen-based network, with the southern side displaying the highest diversity, we didn't find any significant difference both in the H2 index and the connectance in our three sites when we compare the visit-based network, which is the blacker bar here, with the simulated network, which is the light gray bar here. And this was true for both indices and all sites. So this means that the richer communities are not undersampled than poorer communities. To investigate changes in the network structure and in network classifications, I use the black model methodology, who finds the best grouping for plants and insects separately. And this grouping has homogeneous connection patterns. And as in this example here, I have the site of Rie, which is my northern side. And we have 3 separate blocks for the plants and 2 blocks for the insects. So when I compare the changes in the block clustering, I use an alluvial diagram, which is this type of diagram here, where we have the visit network on the left and the pollen network on the right. And the bottom, we have the insect species clusters, and on the top, the plant species cluster. So in these alluvial diagrams, I highlighted in color only the species that actually change their block. And so if we take this site here, we can observe that in the block 2 in the visit network, most parts of species remain in the same block, just one or few species change block in the pollen network. So despite this species rearrangement that we observe in all the 3 sites, we didn't observe any substantial changes in the network structure. And this result was also confirmed by the NMI index, which is a normalized mutual information index, which was close to 1, meaning there were almost a perfect congruence between the visit network and the pollen-based network clusterization. So we don't have any changes in the network structure, but what about species position? So to introduce the concept of species position, I have to introduce the concept of motif based on the work of Simmons et al. 2019. So if we imagine our network as a Lego house here, the motifs are the different pieces with different colors and different sizes that compose the house. And within each motif, we have species position representing the species roles. And in each motif, we have at least 2 positions, 1 position for the plant and 1 position for the insect. So the same species can appear in different positions in different motifs, and the change of position depends on the complexity of the motifs, the number of species, but also on their interactions. So by counting the frequency with which species occur in different positions, we can quantify the species roles, which depends on direct interaction, so the interaction between the group of plants and the group of insects, but also indirect interactions. So the interaction within each group, between pollinators or between plants, which that are competing for the same resource. So as we expected, we found that when we add pollen information, we had changes in the species role, especially for singleton species. And in this example here, we have a singleton plant species, the Achilla millefolium, with only 1 partner and 1 interaction. And so the position in the visit-based network are only one-to-one positions, which means that position with only one partner, as in this pre-example that I pointed. But when we add the pollen information, we observe and we find that there is another partner in interaction with these species. And this means that now is not a singleton species anymore, because now the Achillea has two partners. And a new partner brings also new positions. And so now we can find our species in the simulated position in position with two partners, as in these two examples here. So when we compare the visit position with all the simulated positions, we found that the distance between the visit position and the simulated centroid was greater than the 95% of all the simulated distances, so all the simulated position, which means that there is a change in the species position and in the species form. But this result was not only observed for singleton species, but also for more connected species, such as in Osmia bicolor here, where once again we have a great distance between the visit position and the simulated centroid, and the difference in the species position was not due to the addition of a new partner, but the loss of a partner which was not confirmed by the pollen-based in the pollen-based network. And this result was also true for other sites in my study. And for example, here we have a pollinator species and another plant species that were observed in connection in the visit-based network, but not in the pollen one. So this brings to changes in both species positions. So to conclude, our results show that recording plant-pollinator interactions from visit observation does not provide a biased picture of the network. Use of pollen can provide more complete information at the species level, and this will be especially meaningful for study investigating the pollinator effectiveness, for example. So the take-home message of my study is that when we use visit-based observation, this is a faster and appropriate methodology to gain information on the plant-pollinator interaction network. But a more detailed methodology, such as the pollen analysis, can really improve our knowledge of the species whole within ecological communities, with important implications also for conservation. So thank you for listening, and if you are interested in learning more about our work, you can read our paper, which is published on Ecologica. And thanks for having me in this meeting.
Thanks, Natasha. That was a fantastic talk, and you're very good at keeping time. Lots of time now for a discussion and questions, and yeah.
Natasha, I suspect there's a lot of people breathing a sigh of relief after seeing your results, realizing their visitation networks are still very good.
Oh, I'm glad.
Just waiting to see if some questions come in here. Paolo has a question, so I'll wait a minute for that. Okay, so Paolo's asking, do you find changes in the role of highly connected species across the two net representations?
Yes. Among all the differences in the role position, half of these differences were for singleton species and the other half was for more connected species. And I was expected for singleton species because with the additional pollen, they all increase the number of the interaction, but also for more connected species, sometimes they really increase their interaction with species that we didn't observe before in interaction and visit-based network. So it was a really interesting result.
Cool. And then another question from John Thompson is, how may pollen amount per flower versus pollen distribution among flowers? So some pollinators deposit much more pollen per visit than others. So how could you incorporate that information?
Yeah, so I didn't really quantify the amount of pollen. I only add as a single interaction. And what I did was to use a threshold. So when I count less than 5 pollen grains of one species, I didn't consider that like a true interaction. This was the only difference that we made or tried to make, but we didn't really count the number of grains for different species, but only consider as an interaction or not interaction.
Yeah, that makes sense. And then Anna asks, she's wondering what this means regarding the prevalence or importance of pollen or nectar thieves. So how often do you find species that visited certain flowers but never carried their pollen or their free pollen?
Yeah, so I had this thing for several interactions. So when I separated the information for the two types of pollen, I also separate when I have only an interaction in the observed, so in the visit network and not in the pollen one. And this was also the case for different species. And the major difference was based on the threshold that we decided to use. So even if sometimes there was one pollen grain, we didn't think that was enough to justify the interaction or just to confirm the interaction in the visit network.
Yeah, that makes sense. That's interesting.
But this could change depending on the time of the day, or also maybe sometimes the pollinator was just came out from their nest and maybe they didn't have time to collect the pollen. I can make a difference.
Any other questions?
We still have a few minutes for questions, and while we're waiting for people to think up more, oh, here's one.
There's one more, is how many individuals per pollinator species were collected to quantify pollen plant species?
How many, sorry, individuals per pollinators?
Yeah, exactly.
This depends on our sampling efforts. Sometimes for some species, we only have one individual, and for other species, we have several individuals. And this was different depending on the site where we sample the insects. But yeah, we didn't really, I keep all the insects that carry both collected pollen in their structure and pollen on their bodies. So this also reduced my sample. But to see if there was a difference between the two types of pollen, was one of the characteristics of the studies. So
Sure. All right, well, I don't see any other questions. So thanks, Natasha. That was great.
Thank you.
Thanks for that, Natasha. Before we move on to our next speaker, I just want to remind everyone to maybe take a stretch because it's a long evening ahead of us and we want you all to be completely with us. We will have a break at 5:20, but we have a minute to stretch our neck, arms, get up for a minute. All right. We'll start moving on to our next speaker, David Peede, who's going to tell us about Drosophila santomea. David, if you can share your screen. Excellent. Over to you.
All right, thank you. Hello, my name is David Peede. I am currently a first-year PhD student at Brown University. And today I'll be talking about the work done in my undergrad institution, the University of North Carolina at Chapel Hill, during my time in the Matute lab. And my talk is focused on the evolution of host preference in Drosophila santomea. But first, I want to acknowledge and thank Dr. Dana Matute and Dr. Brandon Cooper for all their work and amazing mentorship that made this project possible. So ecological specialization is a process whereby related species evolve to utilize different subsets of the total niche space available to them. Many groups of insects that rely on plants for food or breeding sites display such a pattern and have evolved to specialize on a small fraction of the total plant species available to them, which is known as plant host specialization. The Drosophila melanogaster species subgroup is a well-suited to study the ecological and genetic basis of host specialization. This group contains 9 described species that have evolved over the last 10 to 15 million years to exploit a wide diversity of ecological niches. 4 of the species within the subgroup that are outlined in blue on this phylogeny, Drosophila melanogaster, Simulans, Morsiana, Yacuba, are considered dietary generalists, while 5 other species outlined in orange, Drosophila tessieri, Seychellia, Orena, santomea, and erecta, are thought to be dietary specialists. However, Drosophila tessieri and Drosophila santomea have never been empirically studied with respect to their genetic or physiological basis of behavioral preference for their respective hosts. For this study, I will focus on the most recently discovered species in the melanogaster subgroup, Drosophila santomea, which is endemic to the high altitude mist forest on the island of Sao Tome, which is this little island shown by this little dot off the coast of Africa. While its sister species, Drosophila yacuba, is found in open and drier habitats at lower elevations on the island of Sao Tome, as well as throughout continental Africa. The motivation for this study came from years of field collections on the island of Sao Tome, where it has been suggested that Drosophila santomea breeds exclusively on the non-toxic endemic fig subspecies Ficus commandiacabra fredeniziana, while its sister species, Drosophila yacuba, is considered a dietary generalist. So with the knowledge that ecological specialization is widespread among insects, we wanted to empirically test if Drosophila santomea has specialized on the endemic fig species from an ecological and genomic perspective. So for my talk today, I will first tell you about the ecological and behavioral work performed on the island of Sao Tome, as well as our classical population genetic analyses to detect positive selection. So first, ecological and behavioral studies. So we first studied the phenological structure of the island of Sao Tome for fruiting trees and Drosophila abundance. Each month, we scored the amount of fruit produced by each tree and the number of Drosophila collected in fly traps. With this information, we aim to identify the proximate fruiting period of each fruiting tree, along with the relative abundance of Drosophila over a 2-year time span. The endemic fig species is outlined in red, shown here, and will be outlined in red for the rest of this presentation. And I want to take a moment here to recognize our collaborator, Lucio Materio, who made this part of our study possible. So here I am showing you the relative abundance on the y-axis, which is just the monthly contribution divided by the yearly total for each of the species. On the x-axis is each month over the 2-year time span, starting in January of 2018 and ending in December of 2019. And the first plot up here are the different fruiting tree species, and the bottom plot are the different Drosophila species at different elevations. And in case you're unfamiliar with the notation, F1 just stands for Drosophila santomea jacoba hybrid. And so here, the data is kind of messy, but don't worry, I'll walk you through it. So here we see that, as expected, most species tend to produce the most fruit during the long fruiting season from February to early June, which I'll outline here. And that some species will continue to produce fruit during the shorter rainy season from October to December, outlined here. But what we are really concerned with is seeing if the relative abundance of Drosophila santomea co-varies with the relative abundance of the endemic fig species. So I will clear all of the data of the species that we are not too interested in. And after running a Spearman's rank correlation test, we in fact do learn that the relative abundance of Drosophila santomea significantly co-varies with the relative abundance of the endemic fig species. So with this knowledge, we next wanted to see if Drosophila santomea is preferentially breeding on the endemic fig species. To test this, we gathered fruit from each of the tree species and recorded the identity of each of the Drosophila species that hatched from each fruit. So in this plot, on the x-axis, I'm showing you the fruiting tree species, and the y-axis, the counts of the hatched flies observed. And the first thing you'll notice is that I originally showed you a lot more fruiting tree species. We excluded fruiting tree species from this figure where the fruits were so small that no Drosophila would even lay their eggs on it. So these are the only fruit species that actually produce Drosophila. And so after running a linear model, we find that the fruit species identity is a statistically significant predictor of breeding sites for each Drosophila species. And post hoc analysis confirms that Drosophila santomea preferentially breeds on the endemic fig species, and that Drosophila jacoba preferentially breeds on Ficus mucoso and Psidium guava. With this information, we next wanted to test if there are any differential fitness effects when Drosophila santomea breeds on other fig species. To assess relative fitness, we performed a viability assay where we reared Drosophila on standard cornmeal medium until they were larvae, where they were then transplanted onto one of the fig species, and the number of larvae that reached sexual maturity was recorded, which is shown here. So on the x-axis is the substrates, which is cornmeal, which is a standard lab medium, and then our 3 different fig species found on the island of Sao Tome, and the y-axis is the percent of larvae that reach sexual maturity. And so after running another linear model, we find that species by substrate interactions is a statistically significant predictor of the relative fitness. And post hoc analysis reveals that Drosophila santomea has the highest relative fitness on the endemic fig species, while Drosophila jacoba's relative fitness is uniform across all fig species. The fact that Drosophila jacoba does not differ in the relative fitness among fig species, yet is rarely found breeding on the endemic fig species, suggests that the higher propensity to breed on the endemic fig species is a derived trait found exclusively in the Drosophila santomea lineage, while the physiological ability to properly complete development on fig species is an ancestral trait to both lineages. Furthermore, we find that Drosophila santomea almost exclusively breeds on the endemic fig species, while also exhibiting a decrease in fitness when artificially introduced to the other fig substrates, which suggests that Drosophila santomea has experienced an adaptive evolutionary pressures resulting in the preference for and strict specialization to the endemic fig species. So I've just showed you that the relative abundance of Drosophila santomea co-varies with the endemic fig species, that Drosophila santomea preferentially seeks out the endemic fig species, and that Drosophila santomea has a higher relative fitness on the endemic fig species, which provides our basis for the investigation of the genetic basis of host preference in Drosophila santomea. So chemoreception in insects is controlled by a diverse set of proteins that includes odorant-binding proteins, chemosensory proteins, olfactory receptors, and gustatory receptors. Adorn binding proteins and chemosensory proteins are small soluble proteins expressed in the sensory organs of insects that bind to distinct hydrophobic adorants and pheromones, which facilitate their transport to olfactory and gustatory receptors that initiate signal transduction in sensory neurons. This set of olfactory genes on the slide has been implicated in a number of cases to drive host specialization in insects and are commonly thought to evolve through positive natural selection. So knowing this, we wanted to estimate the relative evolutionary rates of genes, and so we calculated omega values, which is the ratio of non-synonymous to synonymous substitutions. We first compared the average omega value for each olfactory gene family and compared it to the genomic background to see if any olfactory gene families had significantly elevated omega values. So here on the x-axis, I'm showing you the different gene families. On the y-axis are the omega values, and this is just for Drosophila santomea. And unsurprisingly, we found significantly elevated omega values for all olfactory gene families in Drosophila santomea. This next plot is the same x and y axis, but for Drosophila jacoba. And we also found significantly elevated omega values for all olfactory gene families, except for olfactory receptors. I'm now showing you the two plots essentially concatenated onto one. However, despite seeing elevated omega values compared to the genomic background for the majority of olfactory gene families in both species, we see that the average omega value in Drosophila santomea is significantly higher in all gene families except for chemosensory proteins. These results suggest that on average, Drosophila santomea has experienced more adaptive evolutionary forces than Drosophila jacoba, and is consistent with our hypothesis of host specialization in the Drosophila santomea lineage. We also compared the number of unique genes exhibiting an omega value greater than 1, which is indicative of positive selection, in each species to assess if olfactory genes showed an enrichment of elevated omega values compared to the rest of the genome. This is the same plot as before for Drosophila santomea, and after running a Fisher's exact test, we find more genes with an omega value greater than 1 than we would expect by random chance for all olfactory gene families in Drosophila santomea. Showing you the same plot, but for Drosophila jacoba, we only observe this trend in adorant binding proteins and chemosensory proteins. Again, these results suggest that on average, Drosophila santomea has experienced more adaptive evolutionary forces than Drosophila jacoba. Because of the biases and limitations, though, to results that are reliant on only omega values, we also performed 2 additional tests for selection, which I will not discuss in detail in this talk. Here, though, is an overview of all the direct tests for selection with olfactory gene families with the most support across all measures highlighted for each species. If we take a very strict interpretation where a gene family must exhibit signs consistent with adaptive evolution across all tests, we find that adorant binding proteins and gustatory receptors have the strongest evidence for experiencing positive selection in Drosophila santomea, while chemosensory proteins have the strongest evidence for experiencing positive selection in Drosophila jacoba. Interestingly, there are multiple occasions where we find significant evidence for positive selection in the same olfactory gene families for both Drosophila santomea and Drosophila jacoba, which could suggest that olfactory genes on average are evolving more rapidly among Drosophila species, which also includes evolving for loss of function. Lastly, to actually identify candidate genes underlying the evolution of host specialization in Drosophila santomea, we conducted a relatively standard genome scan for positive selection, which is outlined on this slide, by comparing local levels of diversity to the overall background levels of diversity. Our positive selection scan identified 243 candidate genes, 5 of which are olfactory genes, which are shown here. On this slide, I'm showing you the 5 olfactory candidate genes from our genome scans. And what was really surprising is the fact that we did not see signatures of positive selection for any genes in the OBP19 or OBP22 gene clusters, which have been implicated to underlie host specialization in not only a number of Drosophila species, but in also other insects. These results suggest that the evolution of host preference might be predisposed to involve olfactory genes, but the specific genes underlying specialization differ from species to species. So conclusions. So first, I told you that the relative abundance of Drosophila santomea co-varies with the relative abundance of the endemic fig species on the island of Sao Tome. I then showed you that Drosophila santomea preferentially seeks out the endemic fig species for breeding sites. I then showed you that Drosophila santomea exhibits a higher relative fitness on the endemic fig species. And lastly, I showed you that olfactory genes show signals consistent with adaptive evolution in the Drosophila santomea lineage. And lastly, my acknowledgements. I want to thank Dr. Daniel Matute at UNC Chapel Hill, Dr. Brandon Cooper at University of Montana, Lucio Matiro, the members of the Matute Lab, and the members of the Huerta Sanchez Lab for all of the help and feedback along the way, that NIH for funding this work, the American Society of Naturalists for allowing me the opportunity to present this research. And I'm happy to take any questions.
Thanks, David, for a well-timed talk. We have 5 minutes for questions.
That was great, David. Let me read Mark McPeak's question. He says, nice talk, David. The differences in olfactory gene evolution is really cool, but they must have evolved because of the fitness differences you showed, and those differences are driven by other traits. So what do you think causes the fitness differences among different host fruits?
Yeah, so that's a really interesting, and it's on a case-by-case basis. So a really classic example is Drosophila seychellia, which has specialized on this fruit, which is toxic to other Drosophila species. However, the endemic fig species is non-toxic, so nothing jumps out to mind with this example. But you're exactly right. This idea of specialization involves a whole host of other genes outside these olfactory genes, which we saw when we had elevated levels of omega in the rest of the genome in Drosophila santomea compared to Drosophila jacoba.
And then Peter Ralph is asking on Slack, does the native fig live only on some parts of the island or in some habitats?
Yeah, so it's actually only occurs at the altitudes which Drosophila santomea occurs, which was one of the indicators for the study. However, in the hybrid zones in the Midlands on the island of Sao Tome, the Drosophila jacoba does have access to these endemic figs.
That's cool. Just waiting on any other questions.
While we're waiting, I just want to say that there's some really interesting discussion happening on the Slack channel for this session. So if you haven't yet looked at that, it'd be a good time to have a look. And thanks everyone for contributing.
Yeah, I was saying you could also, that Slack channel will still exist after this session ends, so you can ask and answer questions there as well too. One other comment in the chat, were any of the genes that popped out as adapting particularly surprising? Any plans on follow-ups for functional studies?
Yeah, so one in particular to me was this gustatory receptor 97A, which is a bitter taste signaling in Drosophila larvae. And so this bitter taste signaling is normally a way to tell if your substrate is toxic or not. But this is puzzling because the endemic fig is actually non-toxic. So at first we were wondering, well, why would this be one of the hits? But we see that from my previous slides that Drosophila santomea has a higher relative fitness on the endemic fig species. And actually, the bitter taste signaling is not just necessarily for toxicity or not, but it can differentiate between the fine scale cues of different chemical profiles of different fig species. So that might be one to follow up on, where we're still kind of debating if we want to go down the functional route and see if we can actually knock out any of these genes or do any CRISPR stuff.
Yeah, that makes sense. That's cool. I'm just going to sneak in this one quick question. How do you differentiate between the parental species from the hybrids at the larval stage?
So at the, I mean, we couldn't do it in the field, but we didn't actually need to do it in the field. In the lab, we could obviously do it based off of the known crosses, but in the field, to differentiate between hybrids, the Santomea yacuba hybrids, their abdomen is brown compared to a regular yacuba or Santomea, that kind of opaque color.
Awesome, thanks.
Thanks for that, David. Thanks, Casey. We'll now move on to our next speaker, Sarah McPeek, who will tell us about the theories of natural selection on resource provisioning in pollination mutualism. Sarah, if you could share your screen.
Sure thing. Here we go. Oops, I can't see the, here we go. All right, can everyone hear me and see that? Great, thanks. So thanks everyone. My name's Sarah McPeek. I am a second-year graduate student.
Have focused on change in whole plant.
Oh, hello. Sorry, heard some feedback here. But I'm a graduate student with Butch Brody at the University of Virginia. And it's really been a goal of mine to stand up on stage at Asilomar and present some of my research for several years. And so I'm just going to pretend that I'm there instead of at home in my parents' living room in fuzzy slippers. So in this session, we've heard a lot of different ways that we can think about these species interactions that we study. And I'm going to present a different perspective, which is the perspective of consumer resource theory. And in consumer resource theory, we think about a lot of species interactions as taking place in an energy economy, and that all the species in these interactions are consumers and resources that are exchanging energy. And we can divide these interactions into two main categories based on the effect that one species foraging has on the other species. So in consumer resource antagonisms, the consumer species has a negative effect on the resource abundance, and it has a negative effect on its partner species fitness. But in consumer resource mutualisms, while the consumer species depletes resource abundance, it actually increases the other species' fitness. And the majority of theory work on natural selection in these interactions has focused on antagonisms. And I think part of this bias might be because in natural selection in interactions like antagonisms is direct. Selection on the one species is a direct result of another species' foraging. Whereas in a lot of consumer resource mutualisms, such as pollination and seed dispersal, selection on one species is actually an indirect result of another species foraging. And to illustrate that a little bit more, let's think about how selection is acting on these traits in two interactions between a plant and an insect. So in an interaction like herbivory, obviously the plant's goal is not to be eaten. And so traits that will be selected for are things that will affect how the herbivore is able to consume its tissue. So defense traits, things like that. But in contrast, in a consumer resource mutualism like pollination, the goal is not simply to be eaten, but to be pollinated by the consumer in the process of foraging. And so a lot of plants produce nectar for pollinators, and that's their resource. And those traits will certainly impact the interaction, but so will all of these traits that will influence the consumer's behavior in a way that benefits the plant. So in these consumer resource mutualisms, the resource is actually just one means to that end. And that changes the way that we can think about natural selection on these interactions. But if we want to think about natural selection in a consumer resource framework, it's really essential that we focus on the resources themselves, because it's these resource traits that will impact most directly the ecological dynamics of the interaction. And we want to understand how that interaction between plants and pollinators will generate natural selection on these resource provisioning traits that will then feed back on the ecological dynamics. And so today we're going to do that by building a simple plant-pollinator interaction using a bit of math. So I want you to imagine a plant population in a field that has an abundance of R. And for the simplicity of the math we're about to do, we're going to make a few assumptions about this plant. We're going to say that our species is hermaphroditic, that it's self-compatible, that all individuals only produce a single flower, and that nectar is the primary resource that the plants are using to attract pollinators. So think of something that has a really pretty basic floral design like this magnolia here. Not a lot of the bells and whistles of some flowers that attracts pollinators. So they're coming for the nectar. And we can begin by thinking about the ecology of the nectar itself, because the resource has its own dynamics. And so if you imagine a flower as being essentially a cup for nectar, the plant is going to produce nectar, and we're just going to assume that follows a logistic growth curve. And so what that means is that when the reservoir is completely empty, the plant will produce nectar at the fastest rate, which we'll call ZNPR. And as that reservoir fills up, that rate will decline until we reach the carrying capacity for this cup. And that's ZRV, the maximum nectar reservoir of the plant. But there's actually a second part to this equation, because of course, pollinators are going to impact the amount of nectar that's in the plant's cup at any one time by, doing this. And so we're going to assume that the pollinator has a saturating type 2 functional response on the plant's nectar. And what that means is that as the plant population produces a larger crop of nectar, the pollinators will forage on that nectar at a faster rate, which will saturate at this constant A. And that's essentially because, of course, tiny little insects can't consume infinite nectar. And we can also think about how that nectar is going to impact the pollinators population as its resource, as its food. And so in this equation, the pollinator has a conversion efficiency, B, and that essentially describes how much nectar an individual pollinator needs to consume in order to produce one offspring, one baby. And that's going to scale with the functional response on the nectar, and the pollinator is also going to have a death rate of F. Now we can begin to think about the ecology of the plants, and we'll start by thinking about how producing nectar affects a plant without pollinators. So in the absence of pollinators, we'll say that plant growth follows a simple logistic growth equation, and that these two traits that we're following, ZNPR and ZRV, are going to impact the plant population's growth. And that's going to have two effects. So first, we're going to say that producing the structures to make and hold nectar is costly, and that's going to affect the plant's intrinsic rate of increase as a quadratic function of the values of its trait. And second, we're also going to say that there's basically an energy allocation cost for individuals of producing every drop of nectar. And so anytime you make a drop of nectar, you're allocating energy away from other structures. That's also going to have an impact on your growth rate. And so when this population evolves without pollinators, they actually evolve to lose nectar production altogether, because of course they're paying these costs of producing these structures and making nectar, but receiving no benefit from pollinators. Now, if we add in the pollinators, we add in a third term to this equation that describes the benefit that pollinators are providing to plants that's going to positively impact their growth rate. And we'll say that this benefit will increase as the pollinator population size increases, because as there are more pollinators, they'll be interacting more with the plants and in the process providing a greater benefit by fertilizing those individuals. But that's also going to saturate because, of course, the plants don't have infinite ovules to be fertilized. And so when the pollinators are present, the fitness of a plant with these nectar traits actually becomes a stabilizing function. And the reason why that is because the total fitness of this plant can be partitioned into these to costs of producing nectar and producing the structures that make it, and also the benefit the plant is receiving from interacting with its pollinators. And then lastly, because we're using the per capita fitness form of the plant population growth rate, we can easily convert that into the multivariate breeders equation for these two nectar traits that we're following, maximum nectar production rate and maximum reservoir volume. And we'll assume that these two traits have an additive genetic basis and that they can evolve independently of one another. So now we've set up this ecological interactions and the conditions for natural selection to happen. And we're going to use this model today to ask, how does pollinator ecology affect selection on nectar? And so to do this, we're going to plant identical starting populations of these plants in the same abiotic environment with the same values of costs and benefits from nectar, but with different properties in their pollinators. So imagine something like this. And what I'm going to be showing you in the next couple of slides is the outcome, the evolutionary outcome of a plant that's evolved under these different conditions of natural selection imposed by its pollinators. So what I'm showing you here is a trait surface for one of our traits, the plant's maximum nectar production rate. And each of the intersections along this graph describes the evolutionary outcome of one plant population that's evolved with a certain combination of pollinator properties. And the two properties that we're following here are the pollinator's maximum nectar consumption rate. So that's basically how quickly the pollinators are visiting the plants and eating their nectar. And so on this side, we see that pollinators have very low nectar consumption rates, so they're not visiting the plants as much. Whereas over here, they have high nectar consumption rates. They're visiting them a lot and they're eating a lot of nectar. And then on the second axis here, we have maximum pollinator conversion efficiency, or B parameter. And that measures how quickly the pollinators are converting the energy they consume into offspring. So on this part of the graph, we have pollinators that don't need a lot of nectar in order to reproduce. And on this side, we have pollinators that have a really large energy need in order to reproduce. And what you'll notice is that plants are evolving higher nectar provisioning actually for less efficient foragers and lower nectar provisioning for more efficient foragers. And if we look at our second trait, maximum nectar reservoir volume, which remember I told you can evolve independently, we see the exact same trend. Now, this kind of seems a little bit counterintuitive because don't you think that plants would want to feed more active pollinators more because they'll get greater fitness benefits? But let's think a little more carefully about what nectar does to pollinators. So the first thing we know is that feeding pollinators causes them to forage more. So an evolutionary increase in either one of these traits, the rate at which you make nectar or the amount of nectar that you hold, will affect your standing nectar crop of the population. So how much nectar is in the population at one time. As that increases, we know that pollinators will forage on the nectar at faster rates. Thereby, they'll provide a greater fitness benefit to the plants because they're interacting more often with individual pollinators. And we can also say that plants will earn a greater fitness benefit from producing more nectar for inactive pollinators, thus causing them to interact more than they will for pollinators that are always really active and visiting the plants. But because we know that these traits carry costs, we also find that when the plant evolves to make more nectar, we see a slight decrease in its total population size. And that's compensated by the greater fitness benefit that it's getting from interacting with the pollinators. So the population growth is balancing these costs and benefits of the traits. And if we want to think a little biologically about how we could think about this possible pattern that we're seeing in nature, think about traits that will affect your pollinator's functional response. So I think instantly about things like diet breadth and specialization. If your pollinator eats a lot of things besides a plant's nectar, it might actually be selecting for that plant to make more nectar because the plant earns a higher benefit from causing that pollinator to visit it more. Whereas if your pollinator forage almost exclusively on your nectar, that might actually be selecting for plants to evolve lower provisioning because they don't, essentially they're not needing to work harder in order to achieve a fitness benefit. Now, the second thing we know that nectar does to pollinators is that it actually causes them to reproduce more, because in this model, we're considering nectar to be a primary food source, and you need food to reproduce. So again, increasing these two traits is increasing the standing nectar crop, and that's having the effect of providing more food to pollinators. Thereby, you have more pollinators, and the plant earns a greater fitness benefit because it has more pollinator individuals to interact with. And so what we see is that the plant is actually evolving to make the most nectar when its pollinator population is the smallest. And the reason why the pollinator population is small here is because the pollinator has a low conversion efficiency. So that means that pollinator needs more energy in order to reproduce. And because of that, the plant earns a higher fitness benefit from evolving to increase its nectar production when there aren't many pollinators around to begin with. And so essentially by producing more nectar, the plant population can boost its local pollinator abundance over evolutionary time.
Sarah, you have 5 minutes.
Thank you. Let's think about a couple of pollinator traits that might impact this evolutionary trajectory. So things that are going to impact the numerical response. And something that springs to mind to me is things like sociality. So if you think about the structure of social versus solitary species, social species share the burden of gathering the energy to grow their colony with all of the individuals in their population who are foraging. Whereas solitary species are solely responsible for gathering the energy they need in order to reproduce. And so what we could predict from this model is that social species might actually be selecting for lower nectar provisioning than solitary species, because solitary species require a larger energy budget, and thus their population is bumped up by having more nectar from the plants. And Elizabeth Crone did a really interesting study in 2013 where she increased the floral density in a meadow and saw how that affected the local pollinator populations in the following year. And what she saw is that solitary populations had a huge increase the year after those floral resources were increased, when she didn't see the same thing with social species. So that's some evidence that there might be this ecological mechanism to generate an evolutionary benefit from the plant. We can also think about things like body size, because we know that pollinators often visit plants that have more nectar if the pollinator has a larger body size. And that makes a lot of sense from the pollinator's perspective. But what our model suggests is that plants might actually receive benefits from evolving to provide more nectar to those pollinators who are more needy, because it increases the pollinator's local abundance, thereby providing more benefit to the plants. And so just to sum up really fast, I hope I've made you think a little bit about how natural selection on the provider's resource dynamics is an indirect outcome of consumer foraging in these consumer resource mutualisms like pollination. And mutualist resources in pollination, like nectar, have two effects on the provider's fitness, and they're indirect effects because they're both mediated through the consumer species. So first, there's the trait-mediated indirect effect, where producing more nectar increases the consumer's foraging rate, thereby providing a greater pollination benefit to the plant. And second, we have an abundance indirect effect, where increasing nectar production increases the consumer's population growth rate, thereby also providing a greater fitness benefit to the plant. And lastly, using frameworks like this that are based on traits can generate testable predictions about how these resource traits might evolve in nature. And if you study an interaction that's not pollination, you can think a little bit about how this general framework could apply to your system, because the main thing I really want you to get here is that we can think about the ecological dynamics as defining the evolution of resource provisioning in consumer resource mutualisms. And we can also think about how that can feed back on the ecological interaction itself. And the rest, as with all patterns in nature, is up to ecological context. And with that, I'll thank my collaborators, Mark McPeek and Judy Bronstein, a lot of folks who provided really great comments on writing this paper and developing this model, and thank you for listening. If there's time, happy to take questions.
Great talk. We've got time for one quick question.
I'll ask one really quick question, Sarah. It seems like if you're referring to plants and pollinators, they're probably going to have really similar generation times. Like I have a hard time coming up with exceptions to that, but I can imagine other systems where there might. And how much do you think having these synced up generation time matters? Or do you think like lag effects would affect what's going on there?
Oh, that's really interesting. We didn't think explicitly about time in this interaction. We just thought about how those selective conditions are going to change. But lag, it could certainly affect the pace of change, but I think you'd still eventually come to the same outcome because the same conditions of natural selection are still present. They're just shifted, if that makes sense.
Yeah, that makes sense. Yep.
Thank you.
Excellent. Thanks for that, Sarah. Thanks, Casey. We will move on to our next speaker, the last talk for the first half of this session, Alex Karnish. And Alex will tell us about resource investment trade-off between growth and reproduction in 2 species of Datura. Alex, we can see your screen now.
Great.
Excellent. And over to you.
Great, thank you so much. Hi everyone, I'm Alex Karnish. I'm from the University of Arizona, and I'll be talking about how organisms allocate their resources to life history functions and mutualisms. So over the course of an organism's lifetime, they need to make choices about how to allocate their resources. So this is usually split up between 4 main life history functions: maintenance, growth, defense, and reproduction. And so when they're allocating those resources, they have sets of priority rules which determine the order of allocation to the different life history functions. And we found that in organisms that have indeterminate growth, maintenance has been seen to have the highest priority. And this has been seen in water fleas, clams, butterflies, and spruce trees. So mutualisms are embedded in these life history functions. So mutualisms are crucial for the growth, survival, and reproduction of many species, while also usually being costly when it comes to resource investment. So kind of my big question is, how are mutualist mediated life history functions prioritized when it comes to these priority rules? So I'll be focusing kind of with plants and how they allocate their resources, mostly because they're sessile, so they can't move, which means they're probably more vulnerable to local resource limitation. And for plants, they allocate their resources to growth and reproduction. So for growth, they invest resources to produce roots, stems, and leaves. And then for reproduction, there are kind of 3 main categories where they can invest their resources. The first is seed production, so putting resources into producing pollen and ovules that will eventually become seeds. Pollination, where they put resources into flowers to attract and reward animal pollinators. And then seed dispersal, where they put resources into producing structures that attract and reward animal dispersers. And within this kind of general question about how priority rules factor into mutualisms, I'm interested in how different life history strategies affect how mutualist mediated life history functions are prioritized. Right now, you're probably wondering how we find these priority rules, and this is usually done by manipulating resource availability, either by doing starvation experiments or by having separate treatments where there are different levels of resources available. And then once you set up these treatments, you look for resource trade-offs between low priority and high priority functions in order to kind of see which are the most important and down. So a high priority life history function is one where when resources are reduced, investment into that function doesn't change and the size or magnitude of that function doesn't change. So for example, if we say that growth is a high priority life history function, when organisms are grown in an environment where they have access to fewer resources, those organisms will be the same size and grow to be the same size as those that have more resources. A low priority life history function is one where investment is reduced when resources are reduced. So the size or magnitude of that function is decreased. So if we use growth again, for example, if growth is a low priority life history function, when organisms grow in an environment where there are fewer resources, they'll grow to be smaller, and if fewer resources invested into growth, than organisms that have access to more resources. And then when I'm showing these priority rules, I'm going to be using these resource allocation diagrams. So these are just kind of toy conceptual figures to illustrate the priority of different life history functions. So the total length of the line is the amount of available resources that the organisms can use. And the subsections within represent the amounts allocated to separate life history functions. So here, the gray box is the box that represents the amount allocated to maintenance. So moving on to my life history hypotheses, when we look at annual plants, so they grow over one season, they have one chance to reproduce, I hypothesize that they should prioritize investment into reproduction and producing reproductive structures at the cost of growth. So when we compare plants that are grown in a high resource environment to those that are grown in a low resource environment, investment into growth should decrease, but investment to the different aspects of reproduction, so pollination, seed production, and seed dispersal should remain the same. Moving on to perennial plants, so those that grow over multiple seasons and have multiple chances to reproduce, I hypothesize they should make reproduction a lower priority than growth and maintenance. So when plants are grown in a low resource environment when compared to those in a high resource environment, their investment into the different aspects of reproduction, so pollination, seed production, and seed dispersal, should decrease, whereas investment into maintenance and growth should remain the same. And then finally, I have a hypothesis for plants that are able to reproduce autonomously. So that means that they have flowers that are able to be fertilized from the pollen from their same flower. So I hypothesize that species that are primarily autogamous should make pollination a lower priority because they don't need to really attract and reward animal pollinators in order to produce seeds. So when plants that are primarily autogamous are grown in low resource environments, their investment into pollination should be reduced when compared to plants grown in high resource environments. So to do this, I'm using species within the genus Datura. The Jimson weeds, they're located in the Solanaceae, the tomato family, and there are 18 species in a really well-resolved phylogeny. Right now, I'm just looking at 2 different species, but don't worry, I'm planning on eventually expanding the number of species I'm studying to kind of make broader kind of claims about how life history affects resource allocation. So the 2 species I'm presenting on today are Datura discolor and Datura rhytii. Datura discolor is an annual species. It's relatively small. In nature, when they have plenty of resources here in desert like water, they might grow to maybe 1 meter across. And these plants are primarily autogamous. So they have their anthers located just above their stigma. So before the flowers even open, those anthers will dehisce and release pollen onto the stigma, essentially fertilizing the flower before it even opens. The other species, Datura rhytii, is a perennial species. It's relatively large, so in nature here in Arizona, it might get to be between 2 and 3 meters across when it has plenty of water. And this one can self-pollinate, but is not as primarily autogamous as Datura discolor. It still is able to do it, but doesn't do it as the same high rates that discolor does. Both species are pollinated and eaten by hawk moths and their larvae, and both have seeds with elaiosomes attached. So elaiosomes are nutrient-rich fat bodies that attract and reward ant dispersers. So both these plants are dispersed by ants. Moving on, I grew plants in a greenhouse in sand and had 2 nutrient treatments. One is my high nutrient, so high resource treatment, where I gave them a regular strength solution of Hoagland solution. Hoagland solution is a nutrient solution that contains all the macro and micronutrients plants need in order to survive. And then I have my low nutrient, low resource treatment, where I gave them a quarter strength solution of Hoagland solution. And then the flowers are either hand pollinated or left alone and split into 3 different pollination treatments because I just wanted to see the effects of pollination on seed production. And those are a self-treatment where I hand pollinated the flower with pollen from its own flower, a cross treatment where I hand pollinated a flower with pollen from another plant that was blooming that night, and then an autogamous treatment where I left flowers alone to self-pollinate. So just a quick comparison and an example of how I'll be presenting my data for the rest of the presentation. On the left, I'll always have the annual species Datura discolor, and on the right, I'll always have the perennial species Datura radii. And then within the figures, I'll always have the high nutrient treatment on the left and the low nutrient treatment on the right. And you can see in these pictures that plants in both species that had access to fewer nutrient resources were generally smaller than plants that had a lot more resources. So moving into biomass and the plant's investment into growth, our results are consistent with the annual hypothesis, but not the perennial hypothesis. So Datura discolor, the annual species, when it had fewer resources, it had lower above ground biomass, whereas its below ground biomass was not affected by the treatments. I'm not shocked by the below ground biomass being the same. Datura discolor being an annual species doesn't produce that much below ground biomass to begin with. So Datura discolor's investment into growth, at least for above ground biomass, was reduced when it had fewer resources, which is consistent with our annual hypothesis. Datura radii, our perennial species, when it had fewer resources, reduced both above and below ground biomass, so reduced its investment into growth, which is not consistent with our perennial hypothesis, where we hypothesized that perennial plants would maintain their investment into growth. Moving on to investment into pollination. For Datura discolor, our annual species, not all the measurements are consistent with our annual hypothesis, and they are inconsistent with the autogamy hypothesis. So the number of days until first flower and the size of the flowers was the same in both pollination treatments, indicating that investment into pollination was maintained. But the total number of flowers produced was lower when plants grew in an environment with fewer resources. So it's not completely consistent with our annual hypothesis that annual species would maintain investment into pollination. It is inconsistent with our autogamy hypothesis, where we said plants that are primarily autogamous, which Datura discolor is, would decrease their investment into pollination, which is especially shocking because the flower size remains the same in both treatments. And if this plant ostensibly doesn't need to rely on animal pollinators to get their seeds produced, it's kind of strange that they keep their flowers to be this size. So these are flowers about 16 centimeters long. They're relatively big flowers. Moving on to Datura radii's pollination investment. So this is our perennial species. The results are inconsistent with the perennial hypothesis, but consistent with the autogamy hypothesis. So for Datura radii, the number of days until first flower, the total number of flowers produced, and the size of the flowers is the same in both nutrient treatments, indicating that investment into pollination is the same, rather than being lower, as we had hypothesized with perennial species. But this is consistent with our autogamy hypothesis. So Datura radii is not as autogamous as Datura discolor, so we hypothesized that they would maintain their investment into pollination. Moving on to investment in seed production, our results are consistent with the annual and perennial hypothesis. So Datura discolor, the annual species, pollination treatment had no effect on the number of seeds produced, and nutrient treatment had no effect on the number of seeds produced, which is consistent with our hypothesis that investment into seed production would be maintained in annual species. And then for Datura radii, the perennial species, pollination treatment did not have an effect on seed production, but nutrient treatment did. So when plants had access to fewer resources, they produced fewer seeds, which indicates a reduction in investment to seed production, which is consistent with our perennial hypothesis. So just to summarize the species really quickly, so Datura discolor are annual species. When resources are reduced, investment in growth is reduced, whereas investment in reproduction is maintained, so that its investment into pollination and seed production is the same, and growth is reduced, is consistent with our annual plant hypothesis and inconsistent with the autogamy hypothesis. For Datura radii, our perennial species, when the resources were reduced, investment in growth and seed production were reduced, and investment in pollination was maintained. So this is inconsistent with the perennial hypothesis, where we said that perennial plants would maintain growth and reduce investment into pollination and seed production and other aspects of reproduction, and consistent with the autogamy hypothesis that plants that aren't as autogamous should maintain investment in pollination. Just some take-home messages that I want you to take from this presentation. The main one is that investment in some mutualism-mediated life history functions decreases when resources are limiting. So resource availability does affect mutualism investment. And the pattern of those reductions are related to life history traits. So whether or not a plant is an annual or a perennial, or whether it relies heavily on animal pollinators, can affect resource allocation. And just to kind of look forward, you may have noticed that I didn't talk about seed dispersal investment. Currently, I'm measuring changes in investment to dispersal under resource limitation by measuring the mass of the seeds and eliosomes. And I will start measuring the caloric content of those eliosomes in the near future. And I'm really excited to start looking at that. And I'm currently working on adding more species of Datura to this experiment so I can kind of make broader claims about how life history affects resource allocation. As you can see here, these are the main 6 species that I'm interested in using. And there are a bunch of annual species, more perennial species, different kind of autogamy and selfing rates between those, as well as a group of Datura species that have completely lost an eliosome. So rather than being ant dispersed, they are just kind of passively dispersed. I'm really interested in looking at those species to see the effects of losing a mutualism on allocation of those resources to the remaining mutualisms. And with that, I'd like to thank a bunch of people, my advisor, Judy Bronstein, and my committee, as well as lab members, and especially my undergraduate researcher, Gretchen Gabler, who did a lot of the seed measuring and counting. And with that, thank you for listening.
Thanks so much for that, Alex. And we certainly have time for some questions.
I'll ask a question, Alex, while we're waiting for people to type in questions. I really like this talk, and I like your conceptual model of these boxes and whether they can fluctuate in size and stuff. I'm wondering how, it's assuming all those life histories processes are independent of one another, but you can imagine where they might not be, where like say if you invest a lot of energy in growth, that's going to provide more resources because you gain more light or nitrogen or something. And that might give you more resources to invest in reproduction later. And I'm wondering if that matters or not, to sort of, or if that has any effect on sort of your empirical results.
It definitely matters, especially, I mean, everything in this is connected. So one of the things is the plant, Datura discolor didn't produce as many flowers when it had fewer resources. And that's probably because it wasn't able, it had reduced growth, so it was smaller. So that there's kind of a direct one of like, that limits these amount of places you can put flowers, especially with Datura species. Usually they'll kind of branch and make 2 leaves and then have a bud that turns into a flower. So you don't have enough of these branching points. You're really restricted to the number of flowers you can produce, which kind of is like a concrete thing that I've really noticed. And then for resource acquisition, yeah, it certainly matters. A lot of the stuff with kind of resource allocation has been with animals. So I kind of tend to think like animals only have 1 mouth, whereas with plants, they can put in more resources to make more mouths to get more resources. I'm thinking language right here to be articulate. To kind of combat that, the way I'm kind of doing it is to really, really stress these plants. Because you could imagine if they have enough resources, they can put enough resources to make structures to get more resources. But if you stress them enough, you'll hit this point where if you put resource into resource acquisition, you don't get a positive response. Like you don't gain more resources at some point.
That makes sense.
That's the thing that's perpetually going on in my mind, especially the plants. They can do anything.
Right. Yes. A couple other questions in the chat. Sam Watson asks, have you thought about looking into how investment in root growth changes with availability of resources?
I was trying to grab that by looking at below ground biomass. It's certainly something I could look at, and that I probably will look at, especially with Datura wrightii. I didn't really talk about it, but they do form this really large taproot that they use to store resources for the next season. So certainly it's very important. With these, so for perennial species, I didn't look at the following season just because I wasn't able to keep them in the greenhouse that long. But there are probably some effects on root growth, especially perennial species, on maintaining growth the following season. Though I did see a decrease in below ground biomass. So they are saving resources from they are shunting resources there for the next year instead of putting them in what they have this year. So.
Right. And then one quick question from Elizabeth Moore is, have you thought about looking at other metrics for pollination investment, like insect attraction traits or odors or amount of nectar or other things like that, rather than just flowers?
Yes, so that's kind of another aspect of my dissertation that I'm gonna eventually be doing, is kind of growing these plants under these with different amounts of resources, and then seeing, then bringing moths in and seeing whether they're attracted to them or not, whether having fewer resources changes their level of attractiveness to moths. I did try to measure nectar in this experiment, but for some reason, the plants weren't producing nectar in the greenhouse. I have no idea why. But I'm going to grow more plants and hopefully get answers for those parts of pollination.
Great, awesome.
Excellent. Thanks, Alex, for the great talk. And I would like to just take this opportunity to once again thanks all of our speakers today who've had fantastic presentations and good discussions as well. Thanks everyone for participating. We are officially on break, so please take this opportunity to stretch, hydrate, and refresh. And we'll be back again at what's the time in the US? At 5:40 p.m. And join us again at that point to continue the second half of the session. And there's some very exciting talks in that session as well. So we look forward to seeing you again. All right, welcome back everyone. And we're gonna start the next half of the species interaction session. Our first speaker is Carina Baskett, and she has a very interesting talk title, so I had to go and look at her abstract, and she's going to be telling us about flower color in wild snapdragons and hybrid zones. Carina, can you please share your screen with us?
Yep, is that looking good?
Yep, that is perfect. I will mute myself, and it's over to you now.
Okay, thanks. So hi, I'm Karina. I'm a postdoc in the Barton group at IST Austria, and I wanted to thank the organizers for this virtual meeting. It's my 4th ASN meeting, and I hope that if it's their first, I guess a lot of people it is, that you might join next time that it's in person, that we can all be together in person. And so this talk is my Valentine to the snapdragon flowers. Roses are red, violets are blue. Oh, dragonflower, what do you do? And how do I advance slides? Okay. So a lot of us in evolutionary ecology, we're working on the puzzling wonders of nature. So we want to fit together these puzzle pieces of genotype and phenotype and fitness. And I'm particularly interested in where phenotype and fitness intersect, so natural selection as the correlation between phenotype and fitness, and then what are the ecological drivers of selection, what's causing that selection. And so in some systems, different parts of this puzzle are easier than others. And so for example, in these two mimulus sister species where one is bee pollinated and one is hummingbird pollinated and they have very different flowers, it's I mean, you still have to do work to show it, but it's a little bit more obvious from the get-go what's going on with ecology and phenotype. And so we can fit that part together. And then the really challenging part, at least back 20 years ago when this work was done, was getting at the genetics and doing lots of crosses of populations and QTL mapping to understand what are the genes underlying those all these puzzle pieces. Whereas in the other extreme, there are cases like hybrid zones, which are natural experiments where all of that crossing has already been done. And so in that case, it's much easier to understand what's going on with the selection on phenotypes and maybe even genotypes, but the ecology can be totally mysterious. And that's what I'll be talking about here. So, but first, some more details about hybrid zones. So we have 2 different species, or it can also be 2 different populations that come together and exchange a lot of genes in these hybrid areas. There's a lot of gene flow, except for traits or genes that are showing declines. And so where you have on one side of the hybrid zone, one trait value, and on the other side, another trait value. And you can actually estimate long-term natural selection by looking at the cline width, so how steep is it, and looking at dispersal distance. And so we already have then 3 pieces of our puzzle there. We have selection and phenotype and fitness just by having a hybrid zone with trait clines. And so I'm going to talk about a hybrid zone in Antirainum. This is snapdragons that are native to the Spanish, or native to the Iberian Peninsula, and we work on them in the Spanish Pyrenees. And so we have a hybrid zone here where we have these parapatric subspecies, or you might just want to call them populations, but they have different flower colors, and that's their only distinguishing trait that anyone knows about. And they usually live in different parts of the Pyrenees, but where they meet, hybrid zones are readily formed. And here we have an example where there's a valley that has 2 roads, an upper road and a lower road, and they have the same kind of hybrid zone across both roads. And probably these hybrid zones were formed when people started making roads and railroads. This one was recorded over 150 years ago, so it's pretty stable. Yeah, I think that's what I need to tell you about that. And oh yes, and so then we have this really steep flower color cline in this hybrid zone. It's over only 2 kilometers. And so we know that there's really strong selection on flower color. And we know the genetic basis because people have been using snapdragons as a model system to look at genetics of flower color for a long time. And so you don't have to memorize all these names or anything, but just that there's one gene that controls whether there's yellow or not, and then another gene that controls, well, it's actually 2 very tightly linked genes, but it kind of acts as one that controls whether there's high, medium, or low magenta. And so there are 6 different phenotypes here. It's a pretty simple genetic basis, and that's nice because we can actually use the phenotype to get the genotype, so we don't need to actually genotype them. So that makes it pretty easy to do evolutionary genetics in the system. And so we have a lot of these pieces here, but what is the ecology here? What are the selective agents on flower color? Why doesn't the magenta just march across the valley and invade the yellow, or vice versa? Or why don't they just overlap completely and have hybrids all over the place and yellow and magenta all over the place? And so that's the question I am interested in getting at here. And of course, the first thing you might think of with flowers is that it must be pollinators. And indeed, snapdragons are self-incompatible and they're pollen limited, so pollination is an important step in their life history. However, the major pollinators are constant across the hybrid zone, so we don't have one bee on this side of the valley and another over here. They're the same big bumblebees, and so at least in that sense, there's no differences. And then also color is not a signal of different shape or reward quality. It doesn't, as far as we can tell, it's not associated with other traits or scent or anything like that. And there's some genomic work I don't really have time to get into showing that we think that the flower color regions of the genome are the only ones that are different between the subspecies in this area. So it seems like it's the only thing that's different. So it's not actually that clear that the pollinators should really care about flower color if it's not associated with different reward or shape. And also, furthermore, these awesome papers have come out in the last few years saying that there are lots of non-pollinator agents of selection on floral traits. And there's this great meta-analysis recently. And one thing I really liked about the meta-analysis was they differentiated between different types of traits. And so they found that pollinator selection is weaker for traits dealing with attraction compared to actually like the fit of the pollinator in the flower. And flower color is, of course, an attractive trait. So, so I think what all of this means is that when we're asking this big question of what are the selective agents on flower color, actually what we need to do is first ask the question, what is the ecology of flower color? And go really broad with that question, because we don't know that yet with our hybrid zone. We have all these other pieces, but we don't really know much about the ecology. So with that, I wanted to look at both biotic and abiotic factors of the environment. So I'll start with the biotic observational studies in the hybrid zone. And so here I'm asking whether flower color or hybrid status predicts interaction probability for several interactions. And I'm putting them into these categories of their potential effects on fitness, although we have not measured their effects on fitness. And so first pollination, and here we are looking at visitation, and I would call that a direct effect on reproduction. And then I don't really have time to go into the natural history about all of these, unfortunately, so I'll have to zoom through, but we have seed predation, some specialized Rhinusa weevils. Frugivory, I put a question mark because I'm not totally sure what these little weevils in the flowers are doing, but they seem to be eating the ovules, but I have trouble rearing them. And then more indirect effects on reproduction through florivory, which would maybe affect pollinator visitation. But that one, I wasn't sure which category to put it in, because I think a lot of the times the florivores are also going for the pollen. They're eating the pollen, so that might be more direct. And then nectar robbing, so the same Bombus terrestris that is pollinating is also robbing. And then we have some effects on plant size that could be if flower color is pleiotropic for leaf traits or other defensive traits through herbivory or rust. And so it's a lot of interactions, and for each one, we have measurements of it as a presence or an absence of the interaction on many flowers or fruits or plants, depending on what kind it is. Here's how many. So some of these we have over a few years. And so when you add up all those years and sample sizes, it's quite a few plants and flowers. And these data sets were collected all the way from 2004 to this year. So it's been really great to ask people who've been working on this system, hey, do you have any data about this stuff? And to convert it all to a similar format that we could analyze in the same way. And so I have to thank all these people who've given me their data. And so I'll just show you a summary of the results because I don't have time to get into the details. And so here for each one, there's a symbol for each year that it was measured. So for example, pollinator visitation was measured over 4 years and there was no effect of hybrid status. So that's why it's an X. And there's a few little symbols I'll explain in a second. So what you can see is a lot of X's. So there's a lot of non-patterns here where flower color and hybrid status don't affect these biotic interactions or the probability of the interaction occurring. And so I'll go down this hybrid status column. So we see this was the most exciting one where being a hybrid, it means that there's more florivory. So it's worse to be a hybrid in terms of florivory. Now, whether that really matters for the plant fitness, I don't know. We'd have to look into that. But I think what's really interesting about that is this was one of the stronger effects, and same with this color effect here. And what I think is cool is that even if we're not sure what the effect is on the plants, we should probably expect that the one insect here that's actually eating the flowers would be affected the most by the flower color. So I thought that was pretty neat that those effects were stronger. For herbivory and rust, we actually see that the parents do worse than the hybrids, so that doesn't help explain the maintenance of the hybrid zone. And then these other color effects, they're inconsistent, but just essentially like being yellow is better for pollination, and then for seed predation, it just depends on the year whether magenta is better or worse. And for herbivory, in 1 year, being magenta was associated with higher florivory. So there's a lot of inconsistency here, and but I'm still going to follow up with some more analysis of spatial patterns and try to look at whether flower color frequency, like local color neighborhood, affects these interactions. So getting to our abiotic effects or factors. Here I focused in on drought tolerance, and that's because it's linked to flower color polymorphisms in multiple systems, such as this Linnaeus Disparity, and several others. And that's because anthocyanin pigments play a role in stress tolerance in general. And so, yeah. So, but okay, so we don't have a ecosystem with, it's not a desert, it's not a Mediterranean system with annual severe drought, but I do see that precipitation varies tenfold over 30 years. So it seems like there are times pretty frequently when there's much more and much less precipitation. So it could still be an important selective factor in the area. And so I did a preliminary drought experiment looking at, so I used allopatric crosses because somebody was growing them for something else. And I thought, I'm just going to mess with these plants in the greenhouse and see what happens. One thing I rather like about this experiment is the way that I'm doing the drought is I water the controls when a few of them start wilting. So when the plants tell me that they need water, then I water the controls. And every other time I water the droughts. And so over the course of several of these treatments, they do end up quite stressed out, and I ended up killing quite a few plants, like just the perfect amount, you know, like not all of them, but so not like a dry down experiment where you kill all of them. I killed like an intermediate amount, and that was good. And so I found actually that the F1 hybrids had poor survival, especially compared to the magenta parents. So that's pretty cool and intriguing. To me, this is more intriguing than the results of all the insect stuff. But these are from allopatric populations, and so it could be that stuff other than flower color differs between these populations and is causing these effects. Whereas with our hybrid zone plants, they should be really similar genetic backgrounds and only flower color is different. And so, of course, the next step is that we're doing an experiment with hybrid zone seeds. This is happening right now in the greenhouse. I don't have results yet. They've only gone through one cycle of drought. But at least I can show you cool pictures of anthocyanins being expressed on the underside of the leaves. We're not totally sure why this is, why they did this in the greenhouse, because they don't actually do that very often in the field. It doesn't seem like a very stressful place, but we definitely see a lot of variation in anthocyanins. And so we're excited to see whether that has anything to do with drought tolerance, and whether that has anything to do with flower color. And this work is with Sean Stankowski, who's done some work on drought tolerance and mimulus, and Alex Fuster, who was an intern in our group.
Karina, you have 5 minutes left.
What's that?
That's your 5-minute mark.
Okay, thanks. Perfect. So in conclusion, for my poem, My Valentine, if I asked the snapdragons, what do they do? I think they would say, my functions are many. Yes, it is true. So much data you have, yet hardly a clue. So I feel like even though we do have quite a bit of data, I still don't know very much about the ecology of flower color in this system. But I'm really excited to see what happens with this drought experiment. If we see no effects there, then I'm not exactly sure what my next step will be. I guess trying to look at the fitness effects of florivory and seeing if that's really associated with a lot of pollen theft or not. Or maybe digging in more to better measurements of pollination, because these are pretty rough measurements for sure. So yeah, thanks to lots and lots of field assistance. And I just want to quick plug, if you're looking for a field manager or lab manager, let me know, because Bea is really great and she's on the job market, because we don't have a full-time manager. So any questions? Yeah, thank you.
Thanks, Karina, for this very colorful talk, and good luck with your experiments. We have time for a couple of quick questions.
And thanks for bringing the poetry, Karina. It was very excellent. In the chat, Gaston asked, do you know how the whole genome or other genes not linked with coloration vary along the climb?
Yeah, here, let me find this. Yeah, so this was a study where they did an FST scan along the genome, and so they find that at the scale of the hybrid zone, if you just look a few kilometers apart and look at magenta and yellow that are kind of right outside the hybrid zone, you see that there's really low differentiation across the genome, except for genomic regions that contain flower color genes. And then if you go further away, like not even that far away, 25 kilometers or more, then there's more and more differentiation. So, so there might be interesting things going on with local adaptation that are totally unrelated to flower color if you go further away. But in this area, it seems like the only differences are flower color. Yeah.
And then related to that, Sarah McPeek is asking, she's interested in whether other traits besides flower color could be contributing to the biotic effects you observed in your studies. And do you have any thoughts on traits that are correlated with flower color that might affect animal interactions with hybrids, but perhaps might be more difficult to discern or measure?
Yeah, I think in this system, I'm not sure. So there has been some stuff, or some people have looked at scent in the system and they haven't found any differences in scent. But I can't remember whether that was with the hybrid zone plants or other populations. But I think because of this kind of evidence with the genomic work, it seems like it's probably just flower color here. But of course, there are other traits that are probably impacting the interactions with the insects, like like nectar and things like that. It's just that they're not probably associated with flower color. So it would be a good system to look at selection on other traits, especially because we're working here, we're building a pedigree, and so we have some really interesting ways to measure fitness over a long time using the pedigree. So it would be great to like really dig into other traits and look at how those affect the biotic interactions.
Cool.
Great. Excellent talk. Thanks for that, Karina. Thanks, Casey. Next up, we have Annika Rose Person, and she's going to talk about a very hot topic, which is the effect of early snowmelt on plant-pollinator interactions in alpine systems. Annika, are you able to share your screen with us? And I think you're on mute. Right. Can you say something?
No.
Okay, yeah, we can hear you.
Okay, great.
Over to you now.
Okay, thanks so much. Here we go. Okay. Thanks for that introduction. Yeah, my name is Annika Rose Pearson, and I'm really excited to share my work with you today. So I'll be talking about the effects of early snowmelt and topography on pollination in an alpine subalpine ecosystem. So as you all know, mutualisms are crucial interactions across all different types of organisms. They are critical to the generation and also the maintenance of biodiversity. And insect pollination is one of the most important mutualisms on the planet. Almost 90% of plant species rely on insect pollination to reproduce. And additionally, about a third of human-based food crops require insect pollination. However, global climate change is having effects on all kinds of species interactions across the planet, including on pollination. And climate change can impact pollination in all kinds of ways. I'm going to talk about 3 really briefly. Warming can drive plants and also insects to change their spatial ranges, in particular to shift upward in elevation, but this can cause a spatial shift between plants and pollinators. Additionally, physiological changes can occur that cause plants to produce different types of nectar and volatile organic compounds that attract different groups of pollinators to them. Additionally, phenological shifts, which I'm going to focus on today, can lead to mismatches between plants and pollinators. And just to be clear, phenology is the timing of an organism's life history events. So if, for instance, a plant's phenology is advanced because of climate change and its pollinator's phenology is not mirroring that same magnitude of advance, we may see some sort of mismatch between plants and their pollinators. So I'm interested broadly in how phenological shifts affect alpine pollination. So we know that pollination and seed production influence plant demography and community dynamics in the alpine. And some previous work has shown that there are detrimental effects of early snowmelt on plant fecundity. And this can be via exposure to late spring frosts. However, this has been disputed a little bit recently. Although we do see a lot of like late spring, early summer snow events. I saw a couple of those this summer that did have an impact on some flowers, not necessarily in my subplots, but it definitely is something that happens. Climate change, early snowmelt can also affect plants via extended summer, wherein plants that green up earlier on because of climate change may be experiencing a longer period of drought between snowmelt and those late summer monsoonal rains that help rehydrate the soil. However, we do know that topographic heterogeneity can help drive how communities respond to early snowmelt. And in the alpine tundra specifically, topographic heterogeneity can cause localized soil moisture gradients that can affect the length of the growing season and also the phenology of plants across the landscape. And so because of that, systems like the alpine tundra, which have a very heterogeneous topography, may be buffered against the effects of climate change. And this is just another example from this summer where a tiny shrub caused a huge snowdrift to form, and that lasted about 2 weeks. So even something as small as a shrub can affect where snow accumulates in the alpine. So overall, I had 2 main questions. Number 1, how does early snowmelt affect pollinator visitation rates in an alpine subalpine ecosystem? And number 2, how does early snowmelt affect pollinator diversity in this system? So our study system was located at Niwot Ridge, which is just outside of Boulder, Colorado, just up the hill in the Rocky Mountains. And we had 4 sites there that were located in subalpine and alpine environments that are from 34 to around 3,500 meters in elevation. So that's just at and above tree line. And these sites are in a diversity of topographies with slightly different elevations, but pretty different aspects and concavities, convexities. And at each of these sites, we had 2 40 by 10 meter plots, one of which has experimentally advanced snowmelt and one with unmanipulated snowmelt, which I'll talk more about in a second. And at each site, we had 5 subplots in each treatment type that were 2 by 1 meters in size. And these were also located within diverse topographies within the site itself. So it ended up looking a little bit like this. So we had a 40 by 10 meter plot with experimentally advanced snow melt out date, which I'll be calling my early plot from now on, and also a control plot without any type of experimental manipulation, just adjacent to the early snow melt plot. And within those, we had the 5 2 by 1 meter subplots. So you may be wondering how we advance snowmelt if you're not familiar with Niwot. So a few years ago, Kiara Forrester, Bill Bowman, and a bunch of other amazing folks initiated this project called the Black Sand Project, where we add black sand to the surface of the snow in early spring, which decreases the albedo of the snow and causes snow to melt out up to 2 weeks earlier than in control sites. And this has been shown to affect the phenology of some alpine species, and that work is ongoing. So look out for a paper by Kiara Forrester in the next few months. But as you can see from this image here, it does have a pretty severe effect on snow melt out timing, at least in some sites. So to look at pollinators in these different treatments, I performed at least 10 15-minute pollinator observations in each subplot. And I performed these from when flowers open to when they senesce. And then I collected the first 2 morphospecies of each visitor and recorded the identity of the following visitors. I also collected some information about the environment. So I looked at the number of flowers within subplots and also at the site-wide scale. I also recorded some weather and soil moisture data. And then to examine how topography affects the interaction or affects the effect of climate change on pollination, we calculated aspect, slope, and topographic position index, which effectively tells you how concave or convex an area is. And this can be calculated at either a local or a more broad scale. Okay, so before I get into the real data, I wanted to just share with you kind of an overview of how effective the black sand treatment was on each of the sites. This is really preliminary and super coarse data that I created, and we'll have better, more specific metrics in the next few months, I hope. And this is largely where Chiara Forrester's expertise is. So this is just some basic stuff that I came up with. But so here on the y-axis, we have the difference between peak flowering of the early snowmelt plots and the control snowmelt plots in days. And on the x-axis, we have the different sites. So you can see that some sites, like for instance, site number 3 here in the light green, there was a 24-day difference in peak flowering between the early and the control melt-out sites, treatments. Whereas site 2, there was like very little difference, or at least with this metric, no appreciable difference between the early and the control snowmelt sites. So I'll be referring back to this difference in peak flowering days as we go. So over the course of the summer, I was able to observe around 2,000 total insect visitors, and I collected around 300 of those. So far, I've identified about 95 morphospecies, but I expect that number to increase as I get into the lab and start identifying past morphospecies. And Diptera was the most common order. We had a lot of syrphidae flies, but there were also quite a few lepidopterans, some ants, some coleoptera, and also, of course, bees. So just to get into the data, I wanted to show this graph that shows the number of visits per observation period by the day of year. So on our y-axis, we have the visits per observation period. On our x, we have the day of year. And this is only at site number 1. And the treatments are going to be shown using color. So the blue dots and lines are representing the control subplot or the control treatment, and the red will be representing the early snowmelt treatment. So just looking at this data initially, we can see that the early snowmelt treatment appears to be peaking in insect visitor abundance a little bit earlier than the control snowmelt is. It also appears to be peaking quite higher, and I'm not totally sure why that is, but we're hoping to glean more about that as we go. Now, I want to keep in mind that this is one of the sites where there were 7 days between peak flowering between this early and the control snowmelt treatments. So keep that in mind. And now I'll show you all 4 sites and how visitation changed over time in those sites. So across the sites, we can see a somewhat similar pattern. So at site 3, we can see that early snowmelt visitation is peaking a little bit earlier than control snowmelt visitation. That pattern might be reflected in site 4, but it's not super clear. Keeping in mind that, again, these did have different, the treatment had different effects at these different sites. So to understand this in a more statistical way, I did a GLMER, and I looked at a large number of different variables, environmental variables, as well as these flower variables and the treatment effect. And it turned out that the days between peak flowering, so the effect of the treatment on the sites, was significantly affecting visitation rate, as was the number of flowers in each subplot, as was the day of year's interaction with the days between peak flowering. So this isn't outrageously surprising, and it's something that we can kind of glean from these graphs pretty in a straightforward way. Okay, so to understand pollinator diversity, I went through the same kind of group of graphs and statistics. So here on our y-axis, we have insect visitor Shannon diversity or Shannon entropy. And on our x-axis, we have the day of year. And again, we're showing the control site or the control treatment in blue and the early snowmelt treatment in red. And this is again just for site 1 to start out. So here we don't see that much of a pattern that's caused by the treatment effect. We do see that there's kind of this unimodal shape to the data where diversity is peaking at the middle of the summer, which isn't surprising. And now, again, let's remember that there were 7 days between peak flowering in the site. However, it doesn't seem to be affecting insect diversity all that much. So looking at all of the sites together, once again, we don't really see that much of an effect of treatment. We may see a little bit of an effect at site 4, but we'll see with the stats that doesn't actually come out. And typically we have this kind of unimodal pattern where diversity peaks at the middle of the summer, except for in site 3, which seems to be a bit of an outlier there. And keeping in mind that the treatments did affect these sites differently. So to look at these, how different environmental factors and the treatment affected diversity, we performed a GLMER again, and we found that the day of year, its quadratic term, both had a significant effect on insect diversity. And also the number of subplot flowers significantly affected insect diversity and did so positively. And then interestingly, we found that local scale topographic position index, as well as broad scale topographic position index, significantly affected pollinator diversity. So again, topographic position index is a measure of how convex or concave the landscape is. So it's just a measure of, like, I think it can be associated with how much snow accumulates in an area. So that's something we're going to look more into in the future. So just to go a little bit more in depth and hopefully visualize a little bit better how visitor abundance and visitor diversity is changing over time, I created this little animation. So on the horizontal axis here, I'm going to be showing the number of insect visitors, and on the Y or the vertical axis, we have the individual subplots. On the top here, we're showing the control treatment, and on the bottom in the red, we're showing the advanced snowmelt treatment. And then we're going to be looking at only site 1 first, and it's going to be changing over time. So as the GIF goes on, you'll see how the summer progresses. And the colors are going to be representing different morphospecies of insects and their abundances. So as you can see, there does appear to be an increase in visitation at an earlier period in the early snowmelt treatment than in the control snowmelt treatment. And because we know that from the GLMER that the number of insect visitors were affected by flower number, I also wanted to include how flower diversity and floral number was changing over time. So I've included a panel on the right here that will show you the number and diversity of flowers as they change in the control and the early snowmelt plots over time. So here's that animation. And again, these are changing with, or the colors are representing different species of flowers.
You have 5 minutes, Annika.
Thank you. So not surprisingly, the number of visitors is tracking the number of flowers. As we can kind of see there, it's not super clear, but there does appear to be a connection. And again, the control appears to be peaking later than the advanced. And just to show all 4 sites across the landscape, I'm just going to bring those in right now. We can see that there's quite a bit of dynamism going on, right? Like this site on the top right, site 2 over here, is flowering way earlier than site 4 down here. And there's quite a bit of overlap in the species that are occurring among these sites as well. So it's a really dynamic, exciting system. And so to conclude, we asked, how does early snowmelt affect visitation rate? We found that early snowmelt and its interaction with time appear to affect pollinator visitation rate. We found that the subplot flower number also affected visitation rate. We also asked how early snowmelt affects visitor diversity. And we found that the day of year and the number of subplot flowers does affect visitor diversity, and that local and landscape scale topography also appear to affect visitor diversity. So it's important to note that, that this is preliminary data, but I think that we can say that the early snowmelt did have a significant effect, at least on the insect abundance that were visiting those flowers. And we can also conclude that it may be important to consider topography when we're assessing plant phenology and how pollinators will respond to shifts in snowmelt timing driven by climate change. So as I move forward, I'm working on a structural equation model that will look at how topography, the timing of snow melt out and floral landscape all interact to affect pollinator diversity and abundance. I'm also going to be doing a phylogenetic analysis of pollinators. We're also analyzing the broad pollinator community. I put out a bunch of bee bowls to collect a broad pollinator community across the summer. And I also performed a pollinator exclusion experiment to look at how important pollination is to seed set within the different treatments and across the summer. So I have a bunch of awesome undergrads who are helping me with that remotely. So yeah, thank you everybody. Thanks to my funding sources and everybody at CU Boulder, especially Kiara Forrester, who have been so instrumental to this work, and to my committee at UC Riverside, in particular, Nicole Rafferty and Marko Stasiewicz. So thanks everyone.
Thanks, Annika, for the great talk. We have time for a couple of quick questions.
Awesome.
That's great, Annika. One question in the chat is, does the addition of black sand affect the microclimate or temperature of a site beyond speeding up snowmelt? For example, I can imagine the black sand increasing the soil temperature and or decreasing soil moisture after snowmelt, which might affect plant phenology.
Yeah, that's a really good question. We have done some work looking at how the black sand affects microclimate, and we didn't find any significant effect. So this is going to be a 6-year experiment. So each year we put like a thin layer of the black sand on the surface of the snow. And so the actual accumulation of black sand isn't that much actually on the surface of the soil. But we are cutting it off after 6 years of black sand application, so we don't just get like a huge layer of black sand building up and really strongly affecting microclimate. And additionally, I'd like to note that the black sand is inert, so we found that it doesn't have any impact on soil microbes or soil fertility in any way. Did that answer your question?
Yes, thank you.
Yeah, that's cool. That's really clever. That's cool.
Yeah, I cannot take credit for that, but it's a lot of work too. They haul a lot of pounds, like tons and tons and tons of black sand up there every year.
Any other quick questions? Oh, one more. Amazing talk. I'm wondering, how did you collect insects? I recently learned about the insect vacuum, which is actually a children's toy, but can serve as a useful tool for research.
That sounds like a really cool idea. I've heard of those being used also to like exclude certain insects, kind of like the opposite. But yeah, I just use netting, just nets. And I got to where I could identify morphospecies without netting and collecting things. But typically just netting and putting them in vials and freezing them and bringing them back down the mountain with me.
Okay. Thanks, Annika. Thanks, Casey. We'll now move on to our next speaker, William Wetzel, who is going to be talking about macroevolutionary and global patterns of intraspecific variability in herbivory. William, are you able to share your screen with us?
Yes, can you see it?
Yep, it's working and we can hear you just fine. So over to you now.
Okay. All right, hi everyone, thanks for tuning in. I'm really excited to share the first set of results from a new global collaboration called the Herbivory Variability Network. And I'm looking forward to your feedback on these really fresh results. So actually, I think I'm gonna turn off my video to save my bandwidth here. I don't have a lot. Okay. Can everybody still see the slides?
You're good, yeah.
All right, thanks. Okay. One of the most common observations about plant-herbivore interactions is variability. Herbivory is everywhere, but like many ecological processes, it varies tremendously in space and time across ecosystems, species, genotypes, and even organs within plants. This biological variation has inspired generations of ecologists and evolutionary biologists. Most work on herbivory, however, has focused on understanding differences in mean damage levels, asking what leads a plant to get a certain level of herbivory. This beautifully simple framework has taught us most of what we know about plant-herbivore biology, but it overlooks a key aspect of herbivory. And that is the causes and consequences of the variability itself. Herbivory is indeed variable, but some systems are more or less variable than others. For example, herbivory is distributed among the leaves on the left and very heterogeneous on the right. The traditional focus on mean conditions would explain why a given leaf has a certain level of herbivory, but it overlooks the origins and consequences of the variability itself. We can also frame this from a more statistical perspective, where distributions of herbivory among plants within a species not only have means, they also have variability and skew. We can ask what causes a species to have low variability, high variability, or even high skew in these distributions. Theory tells us that variability and skew are important for population dynamics, the outcomes of species interactions, rates of evolution, superspreading and host parasite dynamics, and many other key processes. Despite the theoretical importance of variability in species interactions, and perhaps because of our tendency to focus on differences between means, we lack basic descriptive data on how variability varies among systems. A fundamental understanding of variability patterns could help us build and test major new hypotheses about herbivory, as well as transform long-standing ones. For example, several major hypotheses deal with how plant-herbivore interactions vary with latitude. We know mean herbivory often decreases with latitude, but does this mean that variability increases with latitude because less herbivory at high latitudes means that herbivory can be more Another major set of herbivory hypotheses posits that being large and abundant makes plants more apparent to herbivores, suggesting they should receive herbivory more consistently. But most tests have instead focused on how mean herbivory differs between apparent and unapparent plants, overlooking the variability itself. To collect the basic data we need to start addressing these questions, several colleagues and I recently formed the Herbivory Variability Network. We're a global network of biologists working to understand how and why plant-herbivore interactions vary across the tree of life and around the world. Our central focus is what leads some systems to have higher or lower variability in their interactions with herbivores. More broadly, though, our goal is to advance how we think about variability in ecology and evolution in general. We're about 18 months old, and we already have more than 200 collaborators from 100+ institutions in 30+ countries. In the first phase of our project, we're focused on collecting the observational data we need to lay the groundwork for a macroecological and macroevolutionary understanding of variability. We have 3 initial questions. Is high variability a common feature of plant-herbivore interactions? What types of plants exhibit higher or lower variability in herbivore damage? And what biotic and abiotic factors correlate with variability in herbivore damage? We're interested in this at 2 scales, among plants within populations and among organs or leaves within plants. At this stage in our network, we're simply quantifying variability in herbivory using a standardized protocol. The essence of the protocol is picking a species in sight, randomly selecting 60 plants, and quantifying herbivory at 3 scales for each plant. These include estimating whole plant percent herbivory, proportion of leaves damage, and percent herbivory for 10 leaves. The key feature of our protocol that sets it apart from past work is the really high sampling intensity within populations, which allows us to estimate variability, skew, and other distributional features. So far, the network has completed 716 surveys on 6 continents, including 474 plant species from 135 plant families. We concluded our first phase of data collection in early November, but amazingly, over just the last 2 months, these two outstanding scientists, Mariah Robinson and Luke Zare, have made so much progress wrangling our enormous data set that we have a few preliminary results to share with you today. So I'm going to use those to address 4 questions. How variable is herbivory? And how does variability in herbivory vary geographically, with plant traits, and across scales? I'm going to summarize variability using 2 metrics. The first is the Gini index, which represents the inequality or variability of distribution. It ranges from 0, perfectly even, to 1, perfectly uneven. It normalizes by the total amount of herbivory, allowing comparisons across systems. The second metric is the residuals of the mean variance relationship. Our data, like most biological data, show a strong relationship between the mean and the variance. So we took the residuals of this and used them as a metric of the amount of variance in herbivory, subtracting out the effect of the mean. Okay, first, just how variable is herbivory? As expected, based on just about every naturalist's subjective impressions of herbivory, it's highly variable. Moreover, the variability itself varies among species. This histogram shows the number of species with different Gini values. The median species had a Gini of about 0.56, and for reference, a Gini of 0.5 means about 10% of plants got more than 50% of all herbivore damage, which is substantially uneven. Many species were far above or below this, which gives us lots of important variation to explain with the network's work. It's hard to visualize the field patterns from these Gini values. So here's a panel of 12 surveys I randomly chose from the dataset. Each one has a histogram that shows the number of plants in the population with different levels of herbivory. And you can see high variability and long right tails are clearly the norm for plant herbivore interactions. And this is significant because theory tells us that this shape has key implications for ecology and evolution, influencing population dynamics, selection, and rates of evolution. Okay, question 2. How do plant traits influence variability in herbivory? We have lots of plant trait data in the pipeline, but right now what we have ready is plant size. Fortunately, a plant size is one of its most important characteristics, and size plays a major role in a pleasingly simple hypothesis about what determines variability in herbivory. That is that variability in the distribution of herbivory among plants depends, or is hypothesized to depend, on the ratio of herbivore meal size to plant size. When plants are small, a fixed meal size either completely wipes out a plant or misses it entirely, resulting in a variable distribution. When plants are large, they essentially average over the variation in herbivory to a greater extent, leading to a homogeneous distribution. This hypothesis is almost embarrassingly simple, but it's a central, though perhaps underappreciated component of Feeney's plant apparency hypothesis, one of the most influential ideas in plant herbivore ecology and evolution. As predicted, amazingly, as predicted by this simple hypothesis, larger plants experienced less variability and greater homogeneity in herbivory. In this figure, each point is one survey from our dataset. The x-axis shows the mean plant size in each survey, and the y-axis shows the Gini variability index, and you can see this strong negative relationship. Small plants get hammered or not, while large plants seem to average out the variation in herbivory. This relationship is just as strong even if we use the mean variance residuals to subtract out differences in mean herbivory. This means that this phenomenon is not driven by how much herbivory plants are getting on average. It's really just about how that herbivory is distributed among plants. This result has major implications for our understanding of the evolution of plant defense, suggesting, for example, that herbivore plant defense should be more common for small plants, so they can turn the defenses on if an herbivore shows up, but not turn them on if there's no herbivore there. And constitutive defenses should be more common for large plants, which should always experience a moderate level of herbivory. Question 3, does variability a number of variables here, but today I'm going to focus on latitude. Latitudinal gradients have taught us an amazing amount about ecology and evolution. Several key latitudinal hypotheses assume differences in heterogeneity between the tropics and higher latitudes. Most empirical studies, however, focus on means of biotic interaction intensities, overlooking the variability itself. Before I show you the variability results, I want to show you that, like other large-scale studies, we found a meaningful but noisy negative latitudinal gradient in the mean level of herbivory. So herbivory is higher in the tropics, but there's also a ton of variation. Okay, now when we put variability in herbivory on the y-axis with the Gini index, we see that plants experience more variability in herbivory at higher latitudes. And here's the real zinger, I think. The gradient in variability appears to be stronger than the gradient in mean herbivory, suggesting that geographic variation in variability may be central to these macro-scale ecological patterns. This means that low latitude plants do not just experience higher mean levels of herbivory, they also experience more consistent herbivory. And high latitude plants do not just experience lower mean herbivory, they also experience it more variably. This phenomenon, I think, could be a potential explanation for why plant defense levels are often found not to follow the latitudinal gradient in herbivory. High latitude plants may experience lower herbivory on average, but the infrequent, very high levels of damage they occasionally do experience may be enough for them to maintain high levels of defense, despite the fact that they have lower herbivory on average. When we use the residuals of the mean variance relationship to account for the latitudinal gradient in mean herbivory, the variability gradient is still present, but weaker, suggesting that part, but not all, of the latitudinal variability gradient is related to differences in mean herbivory, suggesting study the mean and the variance in concert. Okay, last question, number 4. How does variability vary across scales? Essentially, we wanted to know if variability patterns across plants would be similar to or different from variability patterns among leaves within plants. This could tell us something about the scale of plant-herbivore interactions that really matters. So that's what this figure shows here. You can see we found a strong positive correlation between the variance among plants and the variance within plants. In other words, some systems have high variance at both scales, other systems have low variance at both scales. This suggests, to me at least, that the factors that promote variability in herbivory transcend these scales. It also suggests that among plant variability is probably not just a function of plant genetic diversity, because then we would expect within plant variability to be unrelated to among plant variability. So there's something else going on as well. Okay, I'm going to leave you with 3 very preliminary take-home messages. First, our data indicate that variability is an essential feature of plant-herbivore interactions. It's too significant to be left out of how we think about the ecology and evolution of plants and herbivores. Incorporating it into the field's major hypotheses and developing novel hypotheses centered on variability patterns, I think has the potential to advance our understanding of plant-herbivore interactions. Second, focusing on variability revealed that plant size, one of the most fundamental plant traits, one of the most fundamental things about being a plant, just how big they are, plays an outsized role in shaping interactions between plants and other organisms. It just boggles my mind that something so simple as plant size could be so important for shaping these distributions. And perhaps maybe it's a good reminder of how important something as simple as geometry is for setting the stage for species interactions. Third, the factors that influence variability seem to transcend scales within and among plants, which suggests that some systems are simply more variable than others for reasons we hope to discover as we continue diving into our dataset, adding additional analyses, phylogenetic analyses, spatially explicit analyses, analyses of herbivore density, pathogens, reproductive damage, plant traits, and many of the other variables that we have. So stay tuned. I want to thank the HerbVar planning group, which includes Mariah Robinson, Lee Dyer, Phil Hahn, Brian Inouye, Nora Underwood, and Susan Whitehead. We've also recently been joined by these 3 outstanding scientists who are helping us to take HerbVar to the next level and make it a really lasting collaborative network. I also want to thank the members of our 7 topical subgroups. And a big thank you to our coordinators, Mariah and Luke, who've done an amazing job with it. Just a massive amount of organizing, wrangling people, emailing, developing protocols, managing data, and more. Thank you for listening.
Thanks, Will, for this fantastic talk. We have plenty of time for questions.
Ooh, questions coming very quick this time. Will, Sarah McPeek asks, this is a huge, exciting collaboration. I'm wondering if your team is collecting data on herbivore population and trait dynamics, as well as those of the plants. I think it'd be interesting to think about factors that impact herbivore population dynamics could be affecting the patterns of variability we see across these different gradients.
Yeah, I totally agree. A subset of the collaborators have collected herbivore data, especially those who are more entomologically inclined. And so, yeah, we're really interested to see how those patterns match up.
That would be cool. And then Joey Bernhardt asks, says, awesome talk. Does the amount of variability in herbivory vary with spatial scale? And does plant size also vary with latitude?
Yeah, so, oh yeah, those are good questions. We have the data collected in a spatially explicit manner, but we haven't dived into those analyses yet. But I am really excited to see how the variability varies with spatial scale. That would be really exciting. So stay tuned. And so it is known that plants are, in general, are larger at lower latitudes, closer to the equator. And I actually forgot to look if there's a signal of that in our data set. So that would be an important thing to check. Let me think. We don't, I think that signal, I think, is stronger for, especially for trees. And we haven't finished cleaning a lot of the larger mature tree data that we have. So most of that data was sort of up to trees up to maybe 2 or 3 meters tall. So we have some tree species in there, but nothing like this, like maximally sized trees. No canopy trees right now.
Cool, thank you.
Yeah.
Any other questions? We've got a couple minutes.
All right, I'm sure there will be more questions for Will later on. And it's now time to dive into the deception end of the symposium session with Adam Stuckert's talk on the genomics of Müllerian mimicry. Adam, are you able to share your screen with us?
Yes.
All right, and we can hear you well, so I'm going to mute myself and it's over to you now.
All right. Great. Thank you, everyone, for virtually attending my talk. My name is Adam Stuckert, and I'll be talking about some work that I've done both as a PhD student in Kyle Summers' lab at East Carolina University, as well as a postdoc in Matthew McManus's lab at University of New Hampshire, where I'm currently stationed, but still virtually. So if you travel throughout nature and you look, there are a huge variety of signals and modalities that signaling is done in nature. So on the top, we have 2 frogs that have visual signals in their skin. On the bottom left, we have water mites that are using tactile signaling to garner matings. We're all familiar with how birds use auditory signals as a way to maintain territories and also acquire mates. And then also there are signals that occur in modalities that we probably don't think very often of, things like olfactory signaling. And I'm primarily interested in visual signaling. And both of these frogs are using their integument, or their skin, as a kind of classic anti-predator mechanism. The frog on my left is using it to blend into the background. Crypsis, the idea being that predators can't eat them if a predator can't find prey items. The frog on the right has pretty much the opposite end of the spectrum as far as an anti-predator mechanism goes. They have these bright colors and patterns in their skin, really contrasting with the background habitat. And it's coupled with the presence of these defensive toxins in their skin. And this combination is known as aposematism. And aposematic species are those that are both conspicuous and defended. And the really important thing about aposematism is that predators have to learn to avoid a particular phenotype. So they have to associate that appearance with a bad outcome for them. And because there's this component of learning where each predator has to potentially learn to avoid that phenotype, we've long thought that predators should select for monomorphism. So they should drive these populations and potentially species towards a single phenotype to kind of ease the burden of learning across species and especially within populations. But that's not really what we see. If we look in nature, we see a lot of variation across species. For example, in this phylogenetic tree of poison frog family, you can see there's a huge diversity of color and pattern. We see a lot of variation that's occurring across populations. For example, these two ladybird beetle populations, they have discrete color morphs. So we have an example of polytypism. And we also see variation that occurs within populations. This can be discrete, where you have kind of or not discrete, where you have a continuous gradient, or it can be discrete, where you have distinct color morphs or polymorphisms within a population. And so the important thing here is that aposematic colors and patterns are really astonishingly variable, despite what we've kind of historically predicted. And so I'm interested in recently, how is this variation produced? And I've been using this little poison frog that's endemic to Peru. Its name is Rhinitomea imitator. And that specific epithet, imitator, refers to the fact that it's a mimic. So as you travel throughout Peru, there are all of these allopatric mimicry complexes involving imitator. Imitator is the frog on the left in each of these pairs. And all of these are kind of non-overlapping mimicry complexes where imitator has evolved to mimic these established congeneric species. And so imitator historically immigrated into Peru. And then the really cool thing here is that adaptive radiations are powerful tools for exploring evolutionary patterns. And so we set about assembling an imitator genome. We assembled it with PacBio data, polished it with Illumina data, scaffolded it with 10x data and nanopore data, and then eventually annotated the genome with Maker. This slide has exactly 22 words on it, I know because I counted, but it represents literally years of agony of my life. Why is it agony? Because the imitator genome is actually fairly large. Turns out it's about 6.8 gigabase pairs in size. Our final assembly had a contig N50, kind of a measure of genome contiguity of 300,000 base pairs, which isn't phenomenal, but we're relatively pleased with given the size of the genome. And we also have really good genic content. So we have 93% of the expected tetrapod orthologs as determined by a Busco analysis, which means that we've got really, really good genic content, especially for a genome this size. This is pretty analogous to a decent mammalian genome. Now, why are we relatively happy with that somewhat mediocre contiguity? It's because poison frog genomes are actually replete in these repeat elements. They're just everywhere in the genome. So in imitator, we have a 6.8 gigabase pair genome. The repeats are mostly lines and unclassified elements, things that we're still kind of working on further classifying. And Gypsy and Mariner are present, but not overly abundant. Now, why am I highlighting these two, Gypsy and Mariner? Because in the other poison frog that's been assembled and published, Uphega pumilio, what they did is they have a similar sized genome, but this genome is really really just chock full of these two elements, Mariner and Gypsy, almost 1.4 gigabase pairs of it, or like half of a human genome almost. are these two repeat elements. So the cool thing here is that we have approximately the same size genome, we think, between these two species. There's some pretty big differences in the quality of these assemblies as they are right now, but we think that they have evolved very differently, which indicates that there's very little ability for these frogs to stop repeat elements or from proliferating, but they have a different evolutionary history, which is really cool to delve into in the near future. So now that we've assembled this genome, we then decided to leverage it to look at color and pattern production. And we have these 4 mimetic morphs of imitator, and so we really wanted to start there. And the cool thing here is that imitator, which is in this dashed box here, and mimics these 3 congeneric species, 2 morphs of Variabilis on the left, Summersii on the top right, and Fantastica on the bottom right. They have a different evolutionary history. So if you look at the phylogeny of Rhytinomea, imitator here has these really short branch lengths, which is indicative of that adaptive radiation that I talked about. Those species that imitator mimics have much longer branch lengths, so it indicates that those species were established prior to imitator radiating out. And so we think that there are different evolutionary histories there. Imitator diverged more recently and adverged onto established phenotypes that those model species had, which means that these groups of species have different evolutionary histories. And that means that we can examine the production of these phenotypic variations from multiple perspectives. So how do they evolve to have that same phenotype despite different evolutionary histories? Are they the same mechanisms or are they different? So it's a nice, powerful system for that. Now, to start with, we took a gene expression approach to examine color and pattern phenotypes within the system, primarily because with a 6.8 gigabase bare genome, it's really fundamentally impossible right now, fiscally as well as computationally, storage-wise, to do these kind of whole genome approaches. So we took a reduced representation approach, and we examined these 4 mimetic morphs of imitator, 2 morphs of Variabilis that imitator mimics, and then 2 Fantastica morphs that have convergent phenotypes with imitator. I'm sorry, they have the same phenotype as imitator. Imitator adverged. I used the wrong language. And what we did is we looked at gene expression in the skin from whole skin from really early on in tadpole life when they're pretty much brand new into the water, all the way through early frogletdom, or just after they metamorphosed onto the water. And so during that time period, they go from not having any sense of pattern to all the way through a semblance of their adult color and pattern. So they're putting down all those chromatophores and that pigmentation that they would have as an adult that then continues developing after they become a juvenile. And if we look at the gene expression data, what we see, what immediately falls out is that imitator falls out as a clump aside from Variabilis, which is on the left, and Fantastica on the bottom right. So imitator is its own kind of separate grouping on this principal component analysis. And this kind of recapitulates the phylogeny. Variabilis and Fantastica are much more closely related than imitator. And we also see, so that's a small proportion of the variance, 13% principal component 2. But the large part of it is that what we see is developmentally, there's a lot of change in gene expression from young tadpoles all the way through young froglets. And so that is the primary driver in our gene expression data. And when we were looking at elements of color and pattern, particularly of some idea do things in other organisms, which is always a really good starting point when you're doing this kind of work. We saw that there are quite a few interesting genes that fell out. A lot of them, I'm going to highlight some of the cool ones. There are quite a few that were in the melanin pathway, things that are involved in either producing melanophores or involved in melanin synthesis. And 2 really common ones from other literature, particularly mammalian literature, is melanocortin receptor 1 and tyrosinase related protein 1, both of which play a role in melanin synthesis. And so we see that these genes are playing likely a pretty key role here in actual pigmentation and color pattern elements. And so this isn't particularly surprising given that these are our main players in a lot of other taxa, but it's interesting that we're seeing them here again. We also see a lot of evidence that pteridine genes, which are eventually produce yellow and orange pigmentation that gets deposited into chromatophores. We see that these genes are playing a pretty important role, especially between color morphs within species. And so these three genes are especially interesting because some other work that we've done in other species and one or two other published studies in poison frogs seem to continuously highlight genes in this pathway as being really important. These genes consistently fall out as differentially expressed between color morphs or patches of skins. So we think that these are really, really important for producing actual pigment elements that produce different colors in these species. And this is true also in a number of other taxa as well. So a lot of vertebrate lineages, there's a number of pteridine genes that are involved in producing coloration. And to kind of corroborate this, some work by Andrew Rubio and the rest of us, this is currently in review. He found doing some network analyses of different skin color patches that a lot of these genes that I just highlighted fall out as hub genes. So those are genes that are particularly important in gene expression patterns and are likely driving other elements of gene expression. And so we see two of those pteridine genes that I highlighted, PTS and XDH, as well as one of those melanin genes, MC1R, the melanocortin receptor. So this is nice independent evidence that we're on the right track here. And one really interesting thing that we haven't delved too deeply into as far as doing any functional validation or anything like that is the possible role of keratin genes in producing different colors and patterns in poison frogs and potentially vertebrates as a whole. Evan Toomey and a bunch of co-authors in a recent AmNat paper found that the actual platelet thickness of the guanine platelet layers within the iridophores, which are the chromatophores that are responsible for blue and white coloration largely, at least we thought, that the platelet thickness within those iridophores is actually a primary determinant of the color of frogs, kind of regardless of what color these frogs actually are. And so because of this, we also see a suite of genes that are in the keratin family that are differentially expressed within our data set. Amongst these are KRT1 and KRT2, so just standard keratin family genes. And these genes have been seen as important for pigmentation and different color production in a variety of other vertebrates. It's not super common that people see those, but we also see a number of other keratin family genes that are differentially expressed between color morphs and species like KRT10, 18. And so we think that the keratin genes, because they're actually producing keratin in the epidermis, differential expression of that can actually change either the thickness of these iridophore layers or maybe their orientation. And so this is an interesting avenue for kind of future work in functional validation that hasn't really been delved into very deeply by anyone, but we think that this is going to be a really cool avenue of inquiry. And so why do these frogs have such variable colors and patterns? Our gene expression data strongly hints at certain color genes being important, particularly melanin synthesis genes, as well as pteridine genes, but we need some functional validation, some of which is ongoing, and we're hoping to do a lot more soon pending funding. Obviously, this is kind of the first pass at this, and allelic variation is definitely going to be important, especially between these different populations and these different species. And so we have a number of projects ongoing to kind of look at that and a lot more planned. And with that, I'll acknowledge my current lab and funding agencies. Thank you for coming to my talk virtually, and I'll take any questions you may have.
Thanks so much, Adam, for that talk. We definitely have time for questions, so over to you, Casey.
Waiting to see what comes in here.
Can I just sneak one thing in? Totally love the pictures of the frogs. They're so cute.
Thank you. That's really why I'm here.
There's a question. Great talk. Regarding the TE% of the genome between species, do you think the common ancestor lacked TEs or has one replaced the other?
This is a good question, and I don't have the answer to that. So almost certainly there were a lot of transposable elements, just because frogs in general have a lot of them, probably. At least that's what a lot of the data seems to indicate. But we don't know really which came first, the transposable elements or the poison frogs.
That makes sense. Another question from Eva Fisher is, yay, working on beautiful frogs. My question is, if there's a species-specific signature of skin gene expression, but amazing color convergence, how do we begin to understand which gene expression patterns are the relevant ones? And do you think this means there are different solutions for generating the same color pattern?
That's a really good question. And that's what we're hoping to get at. I mean, obviously there are going to be species-specific patterns of gene expression based off of, different evolutionary histories, we think. But they also have a relatively recent common ancestor, and they're all congeneric. And so we think that it's likely that they're just kind of utilizing the same mechanisms across species, but we don't know that as of yet. I'm sure that there's a lot of cases where there are certain genes that are playing a role in pigmentation that are producing maybe the actual pigments like pteridine genes. And then in other cases, it might be, an allelic variant that is producing different pattern elements in a similar manner.
Any other questions for anyone out there?
Okay, well, I guess not. So I'll stop sharing and give whoever is next the chance to get ready. Thank you.
It's a chance to look at that frog picture a lot longer. That was good.
Yeah, I was happy to look at the frog as well, but thanks for that, Adam. Great talk, great photos. Thanks, Casey. We can probably start moving on to our next speaker, Bob Week, and they are going to talk about the coevolutionary arms races and the conditions for the maintenance of mutualism. Bob, you're able to share your screen. That looks great. And if you can say something so we can check your sound, that would be great.
Oh, hello?
Yeah, we can hear you.
Okay, great. All right. Thanks everybody for joining me. This has been a really great meeting so far. Lots of really awesome presentations and discussions. So today I'll be talking about the conditions for the maintenance of mutualistic interactions in the face of coevolutionary arms races. So typically in the past, when I've thought about coevolutionary arms races, my mind typically went to these classic antagonistic examples, which seems that like most coevolutionary arms races would be antagonistic. And so here's a classic example where this garter snake is predating on the newt, and the newt has evolved toxicity, and then the snake has counter-evolved this resistance to toxicity. And so there's this escalatory dynamic for greater toxicity and resistance to that. Another classic example is between this camellia weevil and the camellia fruit. The camellia weevil bores into the pericarp of this camellia fruit to oviposit its larvae. And so there's this coevolutionary dynamic for greater mouthparts of the camellia weevil and thicker pericarps of the camellia fruit. However, these coevolutionary arms races are not restricted to antagonisms. We see them also particularly in pollination-based mutualisms, such as that between this Mogastrinchus longirosatus fly and Laparosa anceps flower down in South Africa. And so we see this fly has this ginormous mouthpart that it uses to insert into this narrow but deep floral nectar tube to retrieve a tiny droplet of nectar at the base of that tube. And by being forced to insert its mouthpart further into the flower, it rubs against the reproductive organs of the flower, thereby transferring pollen. And so the idea here is that the flower is inducing selection on the fly for greater mouthparts, so that the fly has an easier time obtaining nectar at the base of that tube. But at the same time, the fly is inducing selection on the flower for deeper nectar tubes, so that way the probability of pollination is increased. And so my question became whether or not the mutualism can break down, what are the conditions under which this mutualism can break down in the face of this coevolutionary arms race. If the mouthpart of that fly becomes so exaggerated beyond the floral tube of the flower, it might not transfer pollen. It might be better described as a nectar thief rather than a pollinator. And vice versa, if that fly is hardwired to visit these flowers and the flowers evolve a floral tube depth that's longer than the mouthpart of these flies, the flies might not ever be able to retrieve that nectar, but forced to pollinate those flowers every visit. And furthermore, I'm curious about whether this outcome depends on the phenotypic interface. So there's these two classic phenotypic interfaces, the trait differences, which is this bigger is better type situation, which you might think that having a greater mouthpart always confers increased fitness, or having a deeper floral nectar tube always confers increased fitness. But there's potential that there's actually some cost associated with this that's completely due to the interaction. We can see that this fly is actually having a little bit trouble trying to insert its mouthpart into some of these flowers. And so there could be some selection, some sort of stabilizing selection to keep the mouthpart from not evolving too far past the flowers. And so I took a completely theoretical approach to understand the consequences of these two phenotypic mechanisms. And so the trait differences model can be summarized by this figure here. So we see the fly fitness W sub X on the left. It increases as the mouthpart length on the x-axis increases past the floral tube depth on the y-axis. And so the brighter colors here correspond to increased fitness, and vice versa for the flower. Another potential mechanism that can mediate this interaction is this offset matching model. And in this case, it's instead of bigger is always better, fitness is actually maximized at some finite offset from the partner trait value. And so we see for the fly fitness on the left that the mouthpart fitness is maximized when that mouthpart length is this lowercase Greek letter delta greater than the floral tube depth of the flower, and vice versa. The floral tube depth of the flower confers maximum fitness for the flower when it is greater than the mouthpart of the fly by this Greek letter delta. And so to understand whether or not this is a mutualism or an antagonism, we can summarize the interaction by the effects on growth rates. And so for the models I'll be considering, these growth rates, M bar represents the population level growth rate, which is an average across all trait values in that species, decomposes additively into this intrinsic growth rate for that species, and this capital I, which represents the effect of the interaction on the growth rate of that species. And we can do that same decomposition for the partner species Y. And so if those capital I's are in the top right, then we see that the interaction can be considered a mutualism. But if either of them become negative, such as the top left or the bottom right, then this mutualism has collapsed into basically a parasitism. And so the question comes down to how do these interaction effects evolve? So it turns out that under this model, these interaction effects are functions of mean trait values. And so really you can boil the question down to tracking the evolution of these mean trait values. And the evolution of these mean trait values classically can be quantified as this covariance of growth rate and trait value. And so what we need to do is we need to calculate these growth rates from those fitness functions I showed you on those previous slides. And so to do this, we could just take this expected lifetime reproductive output, which I had presented on those previous slides. We want to calculate these instantaneous rate of growth that are functions of trait values. And we can do that by taking this little rescaled limit here. And so before I dive into the models, I just want to provide a little overview of the important parameters involved with these models. So the capital BX and capital BY, those are the strengths of biotic selection, the selection induced by this interaction. And those are going to appear in both the trait differences and the offset matching model. That lowercase Greek letter delta is the optimal offset, which occurs in this offset matching model. And GX and GY denote the amount of heritable variation in phenotypes in each of these species. And you can also distinguish them as the additive genetic variances. And those will also appear in both the trait differences and the offset matching models. So under the trait differences model, we get the dynamics, the coevolutionary dynamics are very simple. In fact, they're constant. We see that the change in X bar over time is just a product of the additive genetic variance of species X and the strength of biotic selection on species X, and similarly for species Y. So these trait values are just going to continue along at constant rates independent of each other, even though their evolution is induced by this interaction. And so as the product of the additive genetic variance and strength of biotic selection differs between these species, the rates at which they evolve will also differ. In contrast, under the offset matching model, we see this additional green term appearing on the right here. And so in addition to the product of heritable variation and biotic selection, we see this offset matching term appearing in the green. And so for the evolution of the mean trait in species X, if that X bar increases past Y bar plus that optimal offset, then the rate of change will actually become negative. And so that creates this sort of stabilizing mechanism where the evolution of species X would actually slow down if we were to halt the evolution of species Y. However, this still leads to indefinite escalatory trait dynamics. And so pictured here are the trajectories of mean traits for both the offset matching and the trait differences models. The trait differences models is pictured here with dashed lines. And so we can see, of course, that they're just trudging along at these constant rates. And this capital delta I have on the right pictures the difference in mean traits between the two species. And you can see that that difference between the mean traits of the two species continues to increase indefinitely as well. In contrast, under the offset matching model, which is the solid curves in the middle there, there's still indefinite trait escalation in both of the species. However, the difference between mean traits of those species converges to a finite value, which is determined by the optimal offset. And so to get back to the question of what are the interaction effects, and therefore when does this mutualism break down? So we can calculate what those interaction effects decompose as under these models. So the interaction effects for species X decomposes additively into the intrinsic benefit of the interaction, which is just the benefit, how much this species benefits from the interaction when the strength of biotic selection is equal to 0. So when there's no biotic selection induced by the interaction. And then on the right, we have the product of the strength of biotic selection and the difference between mean traits. And this holds similarly for species Y. And so based off of the previous slide, we can see that because the mean traits are going to continue to, the difference between the mean traits is going to continue to escalate over time. Eventually, the interaction effects for one of these species is going to become negative. And so therefore the mutualism will eventually dissolve into parasitism under this trait differences model. So on the left plot here, we see the evolution of the interaction effects over time. And we see, sure enough, the species Y in the dashed line, eventually the interaction effect becomes negative. And for species X, it continues to increase over time. And we could also calculate the rate at which this transition occurs, which we have on the right. And that's pictured as the difference between the products of heritable variation and strengths of biotic selection for the two species. And this figure would look symmetric if those products were the same for both species. So in contrast, under the offset matching model, we get a different term for the interaction effects, slightly more complicated. So in the purple, we still have the strength of biotic selection. Now in green reappears this sort of optimal offset term, but squared in this equation. But we also see the expressed variation. So this is the variation that you would go out and measure just by measuring the expressed phenotypes of these species appearing in the red there. And this decomposition holds for both species. And so we could see that as the interspecific variation increases for either species, this mutualism is at risk of collapsing into a parasitism, or if the difference between the mean traits continues to increase, then this mutualism can also collapse into a parasitism. However, we saw a couple slides ago that the difference between mean traits under the offset matching model actually converges. And so because it converges to a finite number, that means for certain levels of biotic selection and certain levels of expressed variation, this mutualism can be preserved under the offset matching model. And so here's two examples of that. On the left is interaction that was initiated as a mutualism. So we have the interaction effects on the y-axis, and so we can see that the interaction effects are both positive in this situation. So it was initiated as a mutualism and it was maintained as a mutualism, even though the mean traits, meanwhile, are continuing to escalate indefinitely. On the right, we see a situation of a novel parasitism actually being transformed into a mutualism due to this offset matching mechanism. And so this is just a very simple little theoretical exploration. There's some important caveats to that. I didn't include the effects of abiotic stabilizing selection, and that would prevent mean traits from escalating indefinitely. I ignored any effects of equal evolutionary feedback, such as with the abundance dynamics of these species, and that could also modulate the strength of selection experienced by these species. And I also ignored any multi-specific interactions, that is interactions with 3 or more species, and this can also provide some conflicting sources of selection, complicating the results. So then how do we interpret the results in light of all these restrictions? Well, I would argue a similar interpretation to how we look at lab and field experiments that are controlling for external variables. By controlling for abiotic stabilizing selection and multi-specific interactions and et cetera, we can focus on the effect of the phenotypic interface, whether or not it is the trait differences or the offset matching, on the outcomes of these mutualistic coevolutionary arms races. And before I finish, I just wanted to discuss real quick some implications for mutualistic networks. And this actually ties back pretty well to a talk by Lucas Camacho, I hope I got that pronunciation right, who opened up this session talking about the role of exploitation in mutualistic networks. And so going back to this system in South Africa, this fly in the flower, the fly actually visits a guild of about 20 other flowers, and in fact has been referred to as a keystone pollinator of that region. And it also visits flowers that seem not very well adapted to visits by it, such as this Babiana thunbergii pictured here. It seems that this fly is just totally robbing the nectar out of this flower without transferring any pollen. And the question becomes, if there's sufficient variation in populations of Babiana thunbergii, then there's potential for adaptation, for evolutionary adaptation in response to interactions with this fly. And as Babiana thunbergii adapts in response to interactions with this fly, this can have indirect coevolutionary effects on other members of this pollination network, such as these malachite sunbirds, which are the primary pollinators for Babiana thunbergii. So the question is whether or not this little fly can actually lead to the indirect evolution of elongated beak lengths in those malachite sunbirds, which I think is very interesting and maybe sounds a little bit far-fetched, but perhaps not, according to some recent work done by Paulo Guilherme et al., which demonstrates that actually indirect effects can drive a very large amount of coevolutionary change in mutualistic networks. So with that, I would like to thank American Society of Naturalists for hosting. This has been really wonderful meeting Scott Neusmer, who co-authored this work with me and was also my PhD advisor, the Institute for Bioinformatics and Evolutionary Studies at the University of Idaho for providing a lot of support for me throughout my PhD, and the NSF as well for providing support. And with that, I'll take any questions.
Thanks, Bob. We definitely have time for questions, and I see they're already rolling in. So Casey, over to you.
Thanks, Bob. That was great. Yeah, so Lucas Camacho is asking, great talk, very impressive work. We should talk more for sure. Did you have any expectation when you insert your model in a 3 or more species system? And so when there's more of a network of interactions?
I have some vague expectations. I think an interesting follow-up project to this that I was thinking about is actually very similar to Lucas's work, but instead of using this trait mismatching model, using the offset matching or a trait differences model, and see how these different phenotypic interfaces can, the frequency of different phenotypic interfaces mediating these interactions changes to structured mutualistic interactions. I think that for these escalatory type dynamics, we might see, particularly for the trait differences, we would see probably increased nestedness because in the trait differences, which is a bigger is better type mechanism, the species with greater trait values can always interact with species of lesser trait values.
That makes sense. That's cool. Bob, as you were describing that model, it occurred to me that it seems like there might be different fitness consequences for the plant and the insect. And you kind of touched on this at the end too, that the fly can go to other flowers if it doesn't match the iris, but the iris, I assume, can only be pollinated by that fly. And so is that built into the model already? Like, does that affect the strength of selection on the plant and the fly?
No, I think that would be included under multi-specific interactions, which I definitely controlled for in this. I basically forced these two. If they couldn't visit each other, then they were doomed to extinction.
Cool.
All right, let's thank Bob again for their talk. And we can move on to start getting Nick's setup for his talk about the survival of brood parasitic young and how it's contingent on host siblings. Sounds very interesting. Nick, are you able to share your screen with us?
Let me see. Yep, I can. Let me get it pulled up here. Okay.
And your audio seems fine, so we'll move it on to you then.
Sounds good. Thanks everybody for sticking around for my talk. I'm super excited to talk to you today about one of my dissertation chapters, looking at support for a Goldilocks principle of host optimality for brood parasites. And so just to get started, I wanted to briefly talk about just introducing people to brood parasitism if it's new to you. So brood parasitism is just a symbiotic relationship between parasites and hosts. Parasites lay their eggs in the nests of hosts, and hosts care for those offspring. And this can occur either within species, which is known as conspecific brood parasitism, or between species, which is heterospecific brood parasitism. This occurs in 3 main groups. It occurs in a species of catfish known as the cuckoo catfish, paper wasps, and then probably most well known is in birds, which is the focus of my talk. And so looking at avian obligate brood parasites, those that don't build their own nests and can only parasitize other species, we see that there are roughly 101 species, which is roughly 1% of all birds, are obligate avian brood parasites. There are at least 7 independent origins across 5 different orders. And these species range from specialists that parasitize only 1 or very few species, all the way up to generalists such as the brown-headed cowbird, which is the focus of today's talk, that can parasitize somewhere around 300 different species. And brood parasites have far-ranging effects on a number of bird species, as they parasitize roughly 17% of all bird species. And they often reduce host species fitness because host parents end up taking care of the brood parasite either along with or in spite of their own offspring. And so for a brood parasitic offspring that doesn't share any genetic relatedness to either its host parents or the other nestlings in the nest after it hatches, it really has 2 options for success. It can either kill its nestmates or it can share the nest with those nestmates. And so within killing, there's a couple different ways that brood parasites do this. They can either directly attack the host nestlings or host eggs. This is seen in honey guides, which use a hook on the end of their bill to tear into either nestlings or the eggs, or through eviction of eggs and nestlings, which is commonly seen in cuckoos. Cowbirds, on the other hand, are nest-sharing brood parasites. And so either some or all of the host nestlings of the host species survive in those nests. And so the question becomes is, when is it beneficial to kill your nestmates versus to share the nest with your nestmates? And what we see with this is that it's really a provisioning trade-off. And so brood parasites that are able to stimulate host parents on their own tend to be more of your natural killers, versus brood parasites that have more of a low stimulative ability, but tend to be more competitive, tend to share the nest with nestmates as a way of soliciting provisions from the host parents, and then just outcompeting the host parents when they bring the food to the nest. And so when we look at common cuckoos are highly stimulative, and their begging is actually considered a supernormal stimulus because it stimulates parents to provision at a rate of 3 great reed warbler nestlings. But we see that there's a trade-off with this in that when cuckoos are forced to be raised in nests with host nestling great reed warblers, their growth rates actually drop. And at least early in development, they receive, in this 4 days in the checked box on the graph on the right, they receive much less provisions than what would be expected by random chance. And so this pattern is pretty well established for cuckoos. When we switch over to looking at cowbirds, on the other hand, what we see is that cowbird growth actually seems to be dependent on host brood size in a different way. So whereas cuckoos actually grew worse with host nestlings in the nest with them, we see that to at least some point, cowbird growth is dependent on host nestlings also being in the nest. But beyond that point, it doesn't seem to be anymore. And so what it appears is happening is that there's a bit of a Goldilocks principle with host species. And I should point out that each black dot in the graph on the left represents a different species. So this is a cross species. This is the pattern that's been shown in the prior literature. And so we see kind of this Goldilocks principle where if the nest has too many nestlings or too few nestlings, it's not as good for growth rates of the brood parasite as kind of an intermediate amount of competition in the number of nestlings that the brood parasite has to compete with. And so this also, what the mechanism behind this seems to be is that it translates to provisioning. So cowbirds, in a sense, seem to be using host nestlings to bring in more food. And then once it comes to the nest, with the graph on the right, what we can see is that cowbirds with that are raised with 2 Phoebe chicks tend to receive a greater amount of the provisions that come to the nest than single cowbird chicks alone. And so it seems to be that there's a provisioning mechanism that cowbirds themselves are not able to stimulate host parents to bring in the food without host nestlings, but once the food is there, they tend to be able to outcompete the host nestlings for that food. And so getting to my study system, we wanted to see if this pattern of, this Goldilocks pattern of growth translates to survival for brood parasites. And so within the nest boxes of prothonotary warblers in the Shawnee National Forest in Southern Illinois, we were able to manipulate brood sizes and essentially weed out any effects of predation or ectoparasitism. And so we put cowbird guards in the nest boxes to avoid predation by other cowbirds that may be coming to essentially get the warblers to reset by killing the brood so that they can turn around and parasitize the brood. We used wire guards around the nest boxes to discourage raptors from coming in and eating the nestlings. And then all of our nest boxes are on greased aluminum conduit poles. So we prevented ground predation from squirrels and raccoons and snakes. And then we changed nesting material on the 4th day after hatching to prevent nest failure due to blowfly ectoparasitism. And that seemed to be effective. I didn't find blowflies after doing that. And so what I ended up doing is I essentially, at hatching, I manipulated the brood sizes to foster warbler nestlings out to unparasitized nest boxes in the system to produce treatments of either 0 host nestlings, 2 host nestlings, or 4 host nestlings, each with a cowbird in the nest box. And then I monitored these nest boxes all the way through development, which is, for a cowbird, it takes about 10 days to fledge after hatching. And so what we find using a quadratic regression is that cowbird survival seems to be highly dependent on brood size, just as patterns of growth rate were. And so we see that survival for seems to be best around 2 host nestlings growing up in the nest with cowbirds. And so we see the survival there is somewhere around 80%. The actual number of kind of ideal brood size is 2.3 host nestlings. And then we see that on either side of that too, huge drop-offs in the probability of survival, with 4 host nestlings being much better than 2 host nestlings, but nowhere near as good as 2 host nestlings. And so we got really curious about this as to kind of what was driving this pattern of survival. And so we looked at a little bit more higher resolution of this data. And what we were able to show is that it looks like the costs of suboptimal brood sizes are paid very early in development for the cowbirds. And so if we look in pink is the 0 host nestling treatment, in green is the 2 host nestling treatment, and in blue is the 4 host nestling treatment. We can see that on day 2 and day 3, there are huge drop-offs in the survival of the 0 host nestling treatments. Those cowbirds did not do very well, but if they survived to day 3, they were able to make it most of the rest of the way out. And I should point out that I cut off the number of days post-hatch for this graph at 8. just because cowbirds can fledge on day 9 and day 10, day 9 or day 10. And so I wasn't able to resolve whether or not they had fledged or died, because the adult warblers sanitize the nest, and so they'll pull the dead nestlings out of the nest. So I cut it off at day 8 because they don't seem to fledge by day 8. And then the other thing we can see is that the 2 and the 4 stay, the 2 host nestling treatment and the 4 host nestling treatment stay pretty competitive for the first 3 days. And then after that, the 4 host nestling treatment drops below the 2 host nestling treatment. And so we see that those costs tend to be paid pretty early in development. Looking at a potential provisioning mechanism, we do see that in the 2 host nestling treatment, cowbirds are fed much better than they are in the 4 or 0 host nestling treatment. With 4 host nestlings, the thought is that competition obviously increases from 2 host nestlings, and so cowbirds are just getting fewer feeds because there are more mouths to feed in that nest. And then in the 0 treatment, for the provisioning rate, the thought is just that the cowbirds don't have the stimulative ability to call in provisions from the parents as they beg. Looking at the proportion of provisions to cowbirds, both in the 2 and 4 host nestling treatment, as the 0 host nestling treatment would be 100% of provisions coming to the nest would be to the cowbird. We see that both are above what would be expected by random chance. But then in the 2 host nestling treatment, it is still higher, and there is a significant difference between those 2 groups. And so this got us thinking about the potential effect of what cowbirds might be doing to avoid being in a suboptimal brood size. And so we looked at this by comparing the 2 host nestling treatment to the 4 host nestling treatment in the number of prothonotary warbler nestlings that were reduced out of the nest, that died throughout the nestling period, because we had controlled for every form of predation and ectoparasitism that we could think of. And what we see is that the number of host nestlings reduced is much greater in the 4 treatment compared to the 2 treatment. And even when we look at this proportionally, still the proportion of host nestlings in the 4 treatment that likely starved out were greater than what we saw in the 2 treatment. And so we think that this shows some pretty good evidence for cowbird-mediated brood reduction, and this seems to be pretty consistent with adaptive niche construction by the cowbirds to increase their survival by reducing the number of host nestlings from what's suboptimal closer to what is more optimal. If you notice, the number, the average number reduced was around 1.6, and so this is pretty close to, if you take 4 minus 1.6, that's 2.4. And what we showed with our probabilities is that the optimal brood size in a prothonotary warbler nest is around 2.3 host nestlings. And so in summary, cowbirds appear to be the engineers of their own broods. We identified a survival cost to variable brood sizes. We showed that this cost is paid early in development. We demonstrated that the likely mechanism for optimal survival is provisioning. And we showed that cowbirds are responsible for reducing their broods to a more optimal brood size. And so with that, I want to thank my PhD advisor, Mark Hauber, other co-authors, Wendy Shelsky, Rebecca Kilner, and Derek Tolman, along with Jeff Hoover, members of the Cowbird Lab, and my other 2 field assistants, Katie Stenstrom and Angela Albury. I'd also like to thank the Department of Evolution, Ecology, and Behavior at the University of Illinois at Urbana-Champaign, the Gann Fellowship through the Department of Education, and the Virtual Asilomar Conference for having me for this talk. And with that, I can take any questions.
Excellent work, Nick. Thanks so much for your talk, and we have plenty of time for questions.
Great job, Nick. I'll let you know if any questions show up here.
Okay.
There we go. From Sarah McPeek, says, great talk. You're finding that cowbird nestling success is dependent on host brood size is intriguing. And it makes me wonder whether this might also impose selection on parents' choice of host nests. So do we see any evidence of cowbird parents preferentially laying eggs in nests with moderate brood sizes? Or do you think the effects of cowbird nestlings are having on their brood size post-hatching would swamp out any selection on the parents' behavior?
So I'll answer the first half of that question first. Cowbirds lay eggs before clutch completion. And so they don't wait until a clutch is complete to make that decision. It's typically when there are 3 or 4 eggs already in the nest that they will lay their egg. But there is wide variation in that, so it is totally possible that they could. I mean, I've had a cowbird lay an egg in a nest that had nothing in it, and then a female warbler came in and started incubating that egg. It was the strangest thing ever this summer. I have no idea why that's the case, but so they will lay kind of at any time, but most of the time it's when there are either 3 or 4 eggs in the nest. And then what was the second half of that question? I'm sorry.
It's kind of a continuation of that, but saying, do you think the effects cowbird nestlings are having on their brood size post-hatching would swamp out any selection on the parents' behavior?
So yeah, I think it could. We haven't done that yet.
Yeah.
But that is a thought. Yeah. Thank you.
And then there's awesome. A couple other questions here. One from Andrew Hendry says, any idea why the different brood parasite species converge on different solutions to the same problem?
Yeah, so the brood parasites are pretty diverse. They kind of cover really a whole half of the avian phylogeny, and they're pretty well spread out across that. So I think, yeah, it just probably stems from that, would be my take on that.
And then one other question from Maria is there any evidence that cowbird nestlings compete more with some of the host nestlings than others?
So working on that right now. That data is being analyzed by a fantastic team of undergraduates. And so hopefully I'll know more about that soon.
That'd be interesting to know. That's cool.
Yeah.
Any other questions? Not seeing any. Thanks, okay. Nick. That was really cool.
Yeah, thank you.
Thanks for the excellent talk there, Nick. Lucky last speaker for the session, Joy Bernhard is our next speaker, who is going to talk about life in fluctuating environments. Joy, can you share your screen with us?
Yeah, let me just get it going here.
Yep, that looks like it's working.
Just give me 1 sec. Okay, there we go.
All right, I will mute myself and it's over to you now.
Okay, yeah, thank you, Manpreet, and thanks to all the organizers for putting this conference together. It's my first ASN meeting and it's been really fun to participate, and I'm really looking forward to the next couple of days. So today, there we go. Today I'm going to talk to you about one of the defining features of life on Earth, environmental variability, and the ways in which living systems across scales, from cells to organisms to populations and communities, cope with environmental fluctuations in order to survive and persist. So nature is variable, and sorry, I'm just trying to get this. Oh, there we go. Okay, nature is variable, and almost every environment on Earth fluctuates over time and space, right? Environmental conditions vary from minute to minute, from day to day, and from season to season. And these patterns of variability differ across biomes, and they differ across the globe. And these patterns of variability are expected to change into the future, increasing in some places and decreasing in others. And so living systems exist in a variable world, and ecosystems are often structured by regular variation in the environment. In this top panel here, what I'm showing you is an example of a lake ecosystem, which is famously known for seasonality and abiotic variation, which influences primary production, the timings of peaks in production, as well as secondary production through spring blooms, fall blooms, and transitions in grazing pressure. And variability can also come from biological processes. So in these bottom two panels, for example, population dynamics that are internal to one species or to the connection between two species, as shown in this links here example, define a situation in which oscillations in populations create variability in the environment that other species and parts of the ecosystem must respond to. And environments, we know environments fluctuate at all scales. So the figure that I'm showing you here on the left is a time series of sea surface temperature off the coast of Norway. And the plot on the right shows the same time series, but now visualized in the frequency domain. And what we can see from this figure is that organisms with different generation times, different body sizes, and other traits will experience the same environment in different ways and respond differently. And so we now know that environmental variability and variation in responses to changes in the environment are fundamental mechanisms that allow biological diversity to exist and to persist, right? So if the environment were homogeneous over time and space, we might find a situation in which one species or a limited set of phenotypes dominates the system, right? They might harvest all the available resources and lead to dominance or competitive exclusion of other species. And much of our work in population and community ecology is actually about understanding the feedbacks between environmental variability and ecological structure. And yet, at the organismal and population levels, environmental variability actually poses a challenge to physiological functioning. And this is because organisms usually have a range of environmental conditions over which they perform well, right? And then if the environment varies over time and moves away from these optimal conditions, performance or fitness declines. And so this raises the question then of how do living systems persist in fluctuating environments? If environmental variability poses a challenge, what are the solutions to living in variable environments, whether they're predictable or unpredictable? And this is a really important question to answer because we expect patterns of environmental variability to change in the future. And I've just shown you that responses to variation are critical to biodiversity patterns. And so understanding how living systems persist in fluctuating conditions is really critical to predicting future ecological states. So what I'm going to do today is present to you a new framework that I've developed with co-authors, which describes a set of mechanisms that operate across biological scales that allow living systems to persist in fluctuating environments. And in this framework, we outline 3 classes of mechanisms that allow living systems to persist in environmentally variable environments. But before I go into those classes of mechanisms, I want to remind you that classically in population ecology, we often think about populations as responding passively or acting as filters of environmental variation. So populations essentially integrate patterns of variation, and they essentially sort of smooth patterns of environmental variables. So in these sets of plots here on the left-hand side will be some time series of environmental variation shown in the gray, and the biological response to that variation is shown in the black. So in contrast to passive responses or simply filtering out environmental variation, I'm going to outline 3 classes of mechanisms that differ. And the first is a class of mechanisms that rely on feedbacks. So feedbacks are probably very familiar to all of us here. They rely on organisms or other living systems, cells, or maybe even populations, responding to changes in their own internal state. And the key kind of defining feature of feedbacks is that since the system is sensing a change in its own internal state, it can only respond after the fluctuation has occurred. And so inherent in feedback mechanisms is an inevitable time lag in the response. So this biological response is responding to the variation in gray, but with some delay. In contrast to feedback is maybe a slightly less familiar class of mechanisms that we call feed forward. And feed forward mechanisms differ fundamentally from feedbacks in that they rely on sensing a change in the environment and a change in self. And so often they're based on some sort of cue or signal that the living system uses to anticipate future environmental conditions. So what this bottom plot shows you is a scenario in which the system is able to respond in time with the environmental fluctuation because it's cued on some other condition that happens in advance of that fluctuation, essentially allowing the organism or some other living system to predict the future environment because of some correlation between a cue and an expected future environmental state. So this type of response or mechanism might seem a little bit counterintuitive because it in some ways kind of defies causality. We often think about organisms not being able to predict the future, but only being able to respond to the present. But actually the reliance on cues essentially allows the system to pull the future into the present and act in the present in anticipation of a future environmental condition. And this is, we argue that this is beneficial because it allows the system to prepare in advance. So the third class of mechanisms that we describe here is what we call general adaptive systems. And these are essentially a combination of feedback and feed forward. Okay, so feedbacks, as I mentioned before, are familiar to many of us. They are reactive processes that respond to changes after conditions have deviated from a set point. So a classic example that might be familiar to many of us is thermoregulation, in which the organism is responding to deviations in internal core body temperature. As that temperature deviates from some set point, the system responds to that deviation either through the constriction or dilation of blood vessels to return it back to that set point. And this return back to a set point enables internal conditions to maintain relatively constant over time, and this is beneficial for physiological functioning. So the key features of feedbacks are that they rely on some internal stimulus or some deviation from some internal state. And they are by definition reactive, meaning that they have to occur after the change is sensed internally. And so there is some time delay. In contrast to feedback is feed forward. And feed forward processes are proactive and they're anticipatory. And so they allow organisms and other living systems to prepare themselves in advance for some future state. And so here I'm illustrating feed forward with an example of diel vertical migration in which copepods here experience variable predation risk at the surface of the water. And this is because predators of copepods are visual predators. And so when it becomes light out every day, predation risk increases because the predators can see their copepod prey. And so these copepods, though, if they can internalize some correlation between some environmental cue, like a change in light availability, and they can then capitalize on a correlation between light availability and predation risk at the surface to move away from the predators, swim down to depth in advance of predation risk being the highest at the surface. So you see that here there's some external environmental cue represented in this little sun that allows the copepod to swim down in advance of this event or this time period of high predation risk at the surface. And so feed-forwards are anticipatory, and they rely on external cues like changes in photoperiod or light regimes, which are correlated with some future selective environment like predation at the surface. But what's critical to understanding feed-forwards is to understand that feed-forwards rely on an internal model which relates some environmental cue to an expected future condition. And this model controls the response of the organism to a cue. And so that's illustrated here in this middle panel. So feed-forward examples are everywhere. And one example that I really like of feed-forward is the dropping of leaves by trees in the fall. And so here, these trees are responding to a change in photoperiod that happens in advance of impending winter conditions. And that allows the trees to drop their leaves before winter comes, and they are going to be exposed to potential frost damage. So dropping of leaves is just one example of a feed-forward mechanism that relies on this correlation between a change in photoperiod and some future winter condition. So feedbacks and feed-forwards often or rarely occur in isolation, and they most often occur together. So that example that I gave you of the feed-forward of the diel vertical migration in copepods might also occur in parallel with a feedback, where copepods can actually feed back. They can swim away from predators after they've physically detected them in their environment. And so, these two types of mechanisms often occur in parallel, but it's useful to distinguish them because we expect these two different classes of mechanisms to change differently as the environment changes. But the key thing here is that all of these mechanisms combined allow living systems like this copepod here to essentially minimize its experience of environmental variation. So if predation risk in the surface in the epipelagic zone varies over time, by moving away from the surface, these copepods can actually minimize their experienced predation risk, and this of course enhances their performance. So how do feed-forwards arise? Well, feed-forwards arise as organisms exploit repeated associations between correlated environmental variables with a time lag to anticipate change. So here's an example that comes from E. coli, which I love, which shows, so if you envision, or if you imagine some sequence of events in an environment that are shown here in these blue bars. These blue bars might seem fairly unpredictable. There's a very wide distribution of time lags between each successive blue event. And the same thing for these gray events. There's also a very wide distribution of time lags, meaning that these two events seem somewhat unpredictable, maybe from the perspective of the organism. But what you can see is that there's a very short time lag between the gray and the blue, meaning that the blue event is almost always preceded very closely by the gray event. And so in the context of the gray event, the blue event is actually highly predictable. Even if the organism, in this case the E. coli, cannot predict the blue event, as long as it can sense the gray event, it knows, or it can predict that a blue event will occur soon after. And so this could be an example. An example of this is a change in oxygen and a change in temperature. E. coli might not be able to sense or predict a change in oxygen in and of itself, but if E. coli can sense a change in temperature, and that temperature is correlated with the change in oxygen, then all the E. coli needs to do is to be able to sense a change in temperature in order to predict a change in oxygen. And this is actually what E. coli use to switch between aerobic and anaerobic metabolism as they go from outside to inside the human gut. And so here, the internal model would be sort of encoded in the metabolic network of the E. coli cells themselves. And so feed-forward processes that rely on correlations between multiple environmental variables and cues are everywhere. It's really easy to find examples in phenology. Circadian rhythms are another example of feed-forward or cue-based processes, as well as the diurnal vertical migrations that I just showed you. And feed-forwards and feedbacks don't only operate within individuals. When communities act as collectives, then feedbacks and feed-forwards can operate across scales, from individuals to populations all the way up to communities. And so this is an example of quorum-sensing bacteria, which use feed-forward to cause collective changes in gene expression and behavior. And in this case, the internal model that enables feed-forward might be the architecture of the quorum sensing network itself. And so more generally, the ways in which living systems sense, communicate, anticipate, and respond to environmental fluctuations, we argue, determines patterns of biodiversity themselves. And it's important to understand these feed-forward mechanisms because human impacts are actually altering the informational milieu and the reliability of cues. So an example that I'm showing you here builds on this previous example of trees dropping their leaves. We have observed that when trees are living beside street lights, which essentially decouple the correlation between photoperiod and temperature, they actually hang on to their leaves much longer, and this exposes them to increased frost damage. So not only that, changes in cues can have ecosystem level impacts.
2 minutes left.
Okay, yeah, thanks. So a change, this is an example of an ecosystem from Alaska where bears feed on both salmon and elderberries. And what's happened here over the last few decades of environmental change is that the salmon prey, which rely on social cues to show up in Alaska, and the elderberries, which rely on some sort of temperature cue, they are shifting at different rates, causing synchrony in the availability of these two resources due to these differential cue patterns, which is actually reducing the time span over which prey are available in the ecosystem and having major implications for the sort of energy pathways through this system. So to conclude, what I've shown you is that organisms and living systems across scales use a combination of feedback and feed-forward mechanisms to survive and persist. And the ways in which living systems and the variety of ways in which these systems anticipate and respond to environmental fluctuations underlies patterns of biodiversity. But human activities are changing the availability and reliability of cues. And so if we want to predict biological responses to global change, we need to study the internal models that organisms use to predict future environments and how these models might adapt to changing selective environments. So with that, I would like to thank my sources of funding, and if there's time, I'll take any questions.
Thanks for that, Joey. Unfortunately, you finished right on the dot, so we don't have time for any questions, but I'm sure there will be questions coming in the Slack channel and the chat. In fact, if people could direct their questions to the Slack channel at this point would be great, because it'd be easier for Joey to answer your questions there instead of in the chat, which will cease to exist once we close the session. Before we close, what I wanted to say was it has been such a fantastic day of talks, such engaging and inspiring talks from all of our speakers, both in the second and the first session today. And I would like to take this opportunity to thank everyone for putting all this effort into this to give us such inspiring. I'm coming out of it very inspired and excited by all the signs I heard today. And I'm really looking forward to continuing this in the sessions tomorrow. On that note, we still have a very exciting plenary from Scott Edwards coming up at 9 o'clock, basically now. So I hope to see you all over there. And